Hello, students. Welcome back to Decode, where biology is decoded into simple, meaningful concepts. This is your trusted audio guide for learning biology the smart and effective way. You're now listening to the NCERT Class 11 Biology Audiobook, Unit 1, Diversity of the Living World, covering Chapters 1 to Chapter 4, along with their complete exercise solutions and key questions. If you've already listened to the individual chapter episodes, this combined audiobook will serve as your perfect companion for quick revision and concept reinforcement. And if you're starting fresh, don't worry. This unit will take you through every essential topic step by step, just as you would study it in class. In this comprehensive audio, we'll explore how life is organized and classified, beginning with the living world and taxonomic categories, moving through biological classification, and uncovering the evolution and diversity within the plant and animal kingdoms. You'll hear each NCERT question explained clearly, along with additional conceptual insights that will help you connect ideas and prepare effectively for both school exams and NEET. So, plug in your earphones, focus your mind, and let's begin decoding Unit 1, Diversity of the Living World, from the simplest microorganisms to the most complex plants and animals. This is Biodecode, and you're listening to Unit 1, NCERT, Class 11 Biology, Complete Exercise Solutions and Key Concepts. This is Biodecode, and you're listening to Chapter 1, The Living World. Introduction to Unit 1, Diversity in the Living World. Biology is the science of life forms and living processes. The living world comprises an amazing diversity of living organisms. Early man could easily perceive the difference between inanimate matter and living organisms. Early man deified some of the inanimate matter, wind, sea, fire, etc., and some among the animals and plants. A common feature of all such forms of inanimate and animate objects was the sense of awe or fear that they evoked. The description of living organisms, including human beings, began much later in human history. Societies which indulged in anthropocentric view of biology could register limited progress in biological knowledge. Systematic and monumental description of life forms brought in, out of necessity, detailed systems of identification, nomenclature, and classification. The biggest spin-off of such studies was the recognition of the sharing of similarities among living organisms, both horizontally and vertically, that all present-day living organisms are related to each other, and also to all organisms that ever lived on this earth, was a revelation which humbled man and led to cultural movements for conservation of biodiversity. In the following chapters of this unit, you will get a description, including classification, of animals and plants from a taxonomist's perspective. Ernst Mayr, born on July 1904 in Kempten, Germany, Ernst Mayr, a Harvard University evolutionary biologist who has been called the Darwin of the 20th century, was one of the 100 greatest scientists of all time. Mayr joined Harvard's Faculty of Arts and Sciences in 1953 and retired in 1975, assuming the title Alexander Agassiz Professor of Zoology Emeritus. Throughout his nearly 80-year career, his research spanned ornithology, taxonomy, zoogeography, evolution, systematics, and the history and philosophy of biology. He almost single-handedly made the origin of species diversity the central question of evolutionary biology that it is today. He also pioneered the currently accepted definition of a biological species. Mayr was awarded the 3 prizes widely regarded as the Triple Crown of Biology, the Balzan Prize in 1983, the International Prize for Biology in 1994, and the Crawford Prize in 1999. Mayr died at the age of 100 in the year 2004. Chapter 1. The Living World. How wonderful is the living world? The wide range of living types is amazing. the extraordinary habitats in which we find living organisms, be it cold mountains, deciduous forests, oceans, freshwater lakes, deserts, or hot springs, leave us speechless. The beauty of a galloping horse, of the migrating birds, the valley of flowers, or the attacking shark evokes awe and a deep sense of wonder. The ecological conflict and cooperation among members of a population and among populations of a community, or even the molecular traffic inside a cell, make us deeply reflect on what indeed is life. This question has two implicit questions within it. The first is a technical one and seeks answer to what living is as opposed to the non-living, and the second is a philosophical one and seeks answer to what the purpose of life is. As scientists, we shall not attempt answering the second question. We will try to reflect on what is living, on diversity in the living world. If you look around, you will see a large variety of living organisms, be it potted plants, insects, birds, your pets, or other animals and plants. There are also several organisms that you cannot see with your naked eye, but they are all around you. If you were to increase the area that you make observations in, the range and variety of organisms that you see would increase. Obviously, if you were to visit a dense forest, you would probably see a much greater number and kinds of living organisms in it. Each different kind of plant, animal, or organism that you see represents a species. The number of species that are known and described range between 1.7 to 1.8 million. This refers to biodiversity, or the number and types of organisms present on Earth. We should remember here that as we explore new areas, and even old ones, new organisms are continuously being identified. As stated earlier, there are millions of plants and animals in the world. We know the plants and animals in our own area by their local names. These local names would vary from place to place, even within a country. Probably you would recognize the confusion that would be created if we did not find ways and means to talk to each other, to refer to organisms we are talking about. Hence, there is a need to standardize the naming of living organisms such that a particular organism is known by the same name all over the world. This process is called nomenclature. Obviously, nomenclature or naming is only possible when the organism is described correctly, and we know to what organism the name is attached to. This is identification. In order to facilitate the study, a number of scientists have established procedures to assign a scientific name to each known organism. This is acceptable to biologists all over the world. For plants, scientific names are based on agreed principles and criteria, which are provided in International Code for Botanical Nomenclature, ICBN. You may ask, how are animals named? Animal taxonomists have evolved International Code of Zoological Nomenclature, ICZN. The scientific names ensure that each organism has only one name. Description of any organism should enable the people in any part of the world to arrive at the same name. They also ensure that such a name has not been used for any other known organism. Biologists follow universally accepted principles to provide scientific names to known organisms. Each name has two components, the generic name and the specific epithet. This system of providing a name with two components is called binomial nomenclature. This naming system, given by Carolus Linnaeus, is being practiced by biologists all over the world. This naming system using a two-word format was found convenient. Let us take the example of mango to understand the way of providing scientific names better. The scientific name of mango is written as Mangifera indica. Let us see how it is a binomial name. In this name, Mangifera represents the genus while indica is a particular species or a specific epithet. Other universal rules of nomenclature are as follows. An. Biological names are generally in Latin and written in italics. They are Latinized or derived from Latin, irrespective of their origin. 2. The first word in a biological name represents the genus, while the second component denotes the specific epithet. 3. Both the words in a biological name, when handwritten, are separately underlined or printed in italics to indicate their Latin origin. Sar. The first word denoting the genus starts with a capital letter, while the specific epithet starts with a small letter. It can be illustrated with the example of Mangifera indica. Name of the author appears after the specific epithet, i.e., the end of the biological name, and is written in an abbreviated form, e.g., Mangifera indica Linn. It indicates that this species was first described by Linnaeus. Since it is nearly impossible to study all the living organisms, it is necessary to devise some means to make this possible. This process is classification. Classification is the process by which anything is grouped into convenient categories based on some easily observable characters. For example, we easily recognize groups such as plants or animals, or dogs, cats, or insects. The moment we use any of these terms, we associate certain characters with the organism in that group. What image do you see when you think of a dog? Obviously, each one of us will see dogs and not cats. Now, if we were to think of Alsatians, we know what we are talking about. Similarly, suppose we were to say mammals. You would, of course, think of animals with external ears and body hair. Likewise, in plants, if we try to talk of wheat, the picture in each of our minds will be of wheat plants, not rice or any other plant. Hence, all these dogs, cats, mammals, wheat, rice, plants, animals, etc. are convenient categories we use to study organisms. The scientific term for these categories is taxa. Here you must recognize that taxa can indicate categories at very different levels. Plants also form a taxa. Wheat is also a taxa. Similarly, animals, mammals, dogs are all taxa, but you know that a dog is a mammal and mammals are animals. Therefore, animals, mammals, and dogs represent taxa at different levels. Hence, based on characteristics, all living organisms can be classified into different taxa. This process of classification is taxonomy, external and internal structure, along with the structure of cell, development process, and ecological information of organisms are essential and form the basis of modern taxonomic studies. Hence, characterization, identification, classification, and nomenclature are the processes that are basic to taxonomy. Taxonomy is not something new. Human beings have always been interested in knowing more and more about the various kinds of organisms, particularly with reference to their own use. In early days, human beings needed to find sources for their basic needs of food, clothing, and shelter. Hence, the earliest classifications were based on the uses of various organisms. Human beings were, since long, not only interested in knowing more about different kinds of organisms and their diversities, but also the relationships among them. This branch of study was referred to as systematics. The word systematics is derived from the Latin words systema, which means systematic arrangement of organisms. Linnaeus used Systema Naturae as the title of his publication. The scope of systematics was later enlarged to include identification, nomenclature, and classification. Systematics takes into account evolutionary relationships between organisms. Taxonomic categories. Classification is not a single-step process, but involves hierarchy of steps in which each step represents a rank or category. Since the category is a part of overall taxonomic arrangement, it is called the taxonomic category, and all categories together constitute the taxonomic hierarchy. Each category, referred to as a unit of classification, in fact, represents a rank and is commonly termed as taxon-te, taxa. Taxonomic categories and hierarchy can be illustrated by an example. Insects represent a group of organisms sharing common features, like 3 pairs of jointed legs. It means insects are recognizable concrete objects, which can be classified, and thus were given a rank or category. Can you name other such groups of organisms? Remember, groups represent category. Category further denotes rank. Each rank, or taxon, in fact, represents a unit of classification. These taxonomic groups, categories, are distinct biological entities and not merely morphological aggregates. Taxonomical studies of all known organisms have led to the development of common categories such as kingdom, phylum, or division for plants, class, order, family, genus, and species. All organisms, including those in the plant and animal kingdoms, have species as the lowest category. Now the question you may ask is, how to place an organism in various categories? The basic requirement is the knowledge of characters of an individual or group of organisms. This helps in identifying similarities and dissimilarities among the individuals of the same kind of organisms, as well as of other kinds of organisms. 1. Species. Taxonomic studies consider a group of individual organisms with fundamental similarities as a species. One should be able to distinguish one species from the other closely related species based on the distinct morphological differences. Let us consider Mungifera indica, Solanum tuberosum tato, and Panthera leo. All the 3 names, indica, tuberosum, and leo, represent the specific epithets, while the first words, Mangifera, Solanum, and Panthera, are genera, and represents another higher level of taxon or category. Each genus may have one or more than one specific epithet representing different organisms, but having morphological similarities. For example, Panthera has another specific epithet called Tigris, and Solanum includes species like Nigrum and Melongena. Human beings belong to the species Sapiens, which is grouped in the genus Homo. The scientific name, thus, for human being, is written as Homo sapiens. Genus. Genus comprises a group of related species, which has more characters in common in comparison to species of other genera. We can say that genera are aggregates of closely related species. For example, potato and brinjal are 2 different species, but both belong to the genus Solanum. Lion Panthera leo, leopard P, pardus, and tiger P. tigris, with several common features, are all species of the genus Panthera. This genus differs from another genus Felis, which includes cats. Family. The next category, family, has a group of related genera with still less number of similarities as compared to genus and species. Families are characterized on the basis of both vegetative and reproductive features of plant species. Among plants, for example, 3 different genera, Solanum, Petunia, and Datura, are placed in the family Solanaceae. Among animals, for example, genus Panthera, comprising lion, tiger, leopard, is put along with genus Felis, cats in the family Felidae. Similarly, if you observe the features of a cat and a dog, you will find some similarities and some differences as well. They are separated into 2 different families, Felidae and Canidae, respectively. Order. You have seen earlier that categories like species, genus, and families are based on a number of similar characters. Generally, order and other higher taxonomic categories are identified based on the aggregates of characters. order being a higher category is the assemblage of families which exhibit a few similar characters. The similar characters are less in number as compared to different genera included in a family. Plant families like Convolvulaceae, Solanaceae are included in the order Polymoniales, mainly based on the floral characters. The animal order Carnivora includes families like Felidae and Canidae. Plus, this category includes related orders. For example, order Primata comprising monkey, gorilla, and gibbon is placed in class Mammalia, along with order Carnivora. That includes animals like tiger, cat, and dog. Class Mammalia has other orders also. Phylum. Classes comprising animals like fishes, amphibians, reptiles, birds, along with mammals constitute the next higher category called phylum. All these, based on the common features like presence of notochord and dorsal hollow neural system, are included in phylum Chordata. In case of plants, classes with a few similar characters are assigned to a higher category called division. Kingdom. All animals belonging to various phyla are assigned to the highest category called Kingdom Animalia in the classification system of animals. The kingdom Plantae, on the other hand, is distinct and comprises all plants from various divisions. Henceforth, we will refer to these two groups as animal and plant kingdoms. The taxonomic categories from species to kingdom have been shown in ascending order, starting with species in figure 1.1. These are broad categories. However, taxonomists have also developed subcategories in this hierarchy to facilitate sounder and more scientific placement of various taxa. Let's understand the hierarchy of taxonomic categories. The arrangement starts from the lowest level, that is, species, and moves upward toward broader groups. The sequence is as follows: species, genus, family order, class, phylum, or division, kingdom. As we move from species to kingdom, the number of organisms included increases, but the similarity among them decreases. For example, species represent closely related organisms, while the kingdom includes a vast and diverse group of living beings. This sequence represents the hierarchical arrangement of classification, where each higher level includes the characteristics of the level below it. Can you recall the basis of arrangement? Say, for example, as we go higher from species, kingdom, the number of common characteristics goes on decreasing. Lower the taxa, more are the characteristics that the members within the taxon share. Higher the category, greater is the difficulty of determining the relationship to other taxa at the same level. Hence, the problem of classification becomes more complex. Table 1.1 indicates the taxonomic categories to which some common organisms, like housefly, man, mango, and wheat, belong. Let's look at some examples of organisms and their taxonomic classification. First, man. The common name is man. The biological name is Homo sapiens. The genus is Homo. The family is Hominidae. The order is Primata. The class is Mammalia. And the phylum is Chordata. Next, housefly. The common name is housefly. The biological name is Musca domestica. The genus is Musca. The family is Muscidae. The order is Diptera. The class is Insecta. And the phylum is Arthropoda. Now, mango. The common name is mango. The biological name is Mangifera indica. The genus is Mangifera. The family is Anacardiaceae. The order is Sapindales. The class is Dicotyledonnae. And the division is Angiospermae. Finally, wheat. The common name is wheat. The biological name is Triticum aestivum. The genus is Triticum. The family is Poaceae. The order is Poales. The class is Monocotyledonnae, and the division is Angiospermae. These examples show how every organism can be systematically classified from its common name to its phylum or division. Summary. The living world is rich in variety. Millions of plants and animals have been identified and described, but a large number still remains unknown. The very range of organisms in terms of size, color, habitat, physiological and morphological features make us seek the defining characteristics of living organisms. In order to facilitate the study of kinds and diversity of organisms, biologists have evolved certain rules and principles for identification, nomenclature, and classification of organisms. The branch of knowledge dealing with these aspects is referred to as taxonomy. The taxonomic studies of various species of plants and animals are useful in agriculture, forestry, industry, and in general for knowing our bioresources and their diversity. The basics of taxonomy, like identification, naming, and classification of organisms, are universally evolved under international codes. Based on the resemblances and distinct differences, each organism is identified and assigned a correct scientific/biological name comprising 2 words as per the binomial system of nomenclature. An organism occupies a place or position in the system of classification. There are many categories, ranks, which are generally referred to as taxonomic categories or taxa. All the categories constitute a taxonomic hierarchy. Just finished listening to Chapter 1, The Living World. This is Biodecode, and you're listening to Chapter 1, The Living World, Exercise Solutions and Key Questions. Question 1. Why are living organisms classified? Living organisms are classified to make the study of the vast diversity of life forms easier and more systematic. Millions of species exist on Earth, differing in structure, function, and mode of life. Classification helps biologists to 1. Organize and simplify study by grouping organisms based on similarities. 2. Identify relationships among different species. 3. Understand evolutionary connections, showing how organisms have evolved from common ancestors. 4. Enable easier communication among scientists across the world using a universal system of naming. In short, classification brings order to diversity and helps in studying living organisms more efficiently. Question 2. Why are classification systems changing every now and then? Classification systems change because our understanding of life and evolution keeps improving. With advances in molecular biology, genetics, and phylogenetic studies, new information about organisms' relationships emerges. For example, DNA sequencing has revealed evolutionary connections that were not visible through morphology alone. Therefore, as new data become available, scientists revise the classification system to reflect a more accurate picture of evolutionary relationships. Question 3. What different criteria would you choose to classify people that you meet often? People can be classified using many observable or measurable criteria, such as 1. Physical features, height, complexion, or eye color. Age groups, children, adults, or elders. 3. Gender, male, female, or others. 4. Occupation or profession, students, teachers, doctors, etc. 5. Region or language, based on the place they belong to or the language they speak. This simple exercise shows how classification depends on the purpose and observable characteristics, just as scientists classify living organisms based on shared features. Question 4. What do we learn from identification of individuals and populations? Identification helps in recognizing an organism and assigning it to a known taxonomic group through identification. 1. We understand an organism's distinct characteristics. 2. We can determine its relationships with other organisms. 3. We learn about its ecological role, distribution, and evolution. 4. In population studies, identification helps in tracking biodiversity, conservation efforts, and ecological balance. Thus, identification is the first step in taxonomy and forms the basis for scientific study of biodiversity. Question 5. Given below is the scientific name of mango. Identify the correctly written name. Mangifera indica or Mangifera indica in italics. Correctly written scientific name is Mangifera indica. In binomial nomenclature, the first word Mangifera represents the genus and always begins with a capital letter. The second word indica represents the species and always begins with a small letter. Both words are italicized when printed or underlined separately when handwritten. Hence, Mangifera indica is the correct scientific name for mango. Question 6. Define a taxon. Give examples of taxa at different hierarchical levels. A taxon plural. Taxa is any unit or category used in the biological classification system. Each level in the taxonomic hierarchy, such as kingdom, phylum, class, order, family, genus, or species, is called a taxon. Examples: species, Homo sapiens; genus, Homo; family, Hominidae; order, primate; class, Mammalia; phylum, Chordata; kingdom, Animalia. Each represents a taxonomic rank that groups organisms with shared characteristics. Question 7. Identify the correct sequence of taxonomical categories. The correct sequence of taxonomical categories is species, genus, family, order, class, phylum, kingdom. Among the given options, the third option, species, genus, order, phylum, is the closest to the correct order, but it misses family and class in between. So remember, hierarchy moves from smaller to broader groups. Question 8. What does the term species mean? The term species is defined as the basic unit of classification. In general, a species includes a group of individuals that 1. Share similar characteristics, 2. Can interbreed among themselves, and 3. Produce fertile offspring. In higher plants and animals, species are usually defined by reproductive isolation, meaning individuals of one species do not interbreed with those of another. However, in bacteria and other asexual organisms, species are identified based on morphological, biochemical, and genetic similarities, since they reproduce asexually. Thus, the concept of species varies slightly depending on the organism's mode of reproduction and structure. Question 9. Define phylum, class, family, order, and genus. Let's understand each one. Phylum, a group of related classes that share common fundamental characteristics. Example, phylum Chordata includes animals with a notochord, such as mammals, birds, and fishes. Class, a group of related orders. Example, class Mammalia includes orders like primates and carnivora. Order, a group of related families. Example, order Carnivora includes families like Felidae cats and Canidae dogs. Family, a group of related genera that share several common traits. Example, family Felidae includes genera Panthera lion, tiger, and Felis cat. Genus, a group of closely related species which have common features and are believed to have evolved from a common ancestor. Example, genus Panthera includes Panthera leo lion and Panthera tigris tiger. Question 10. Illustrate the taxonomical hierarchy with examples of a plant and an animal. Example 1. Animal, human being. Kingdom, Animalia. Phylum, Chordata. Class, Mammalia. Order, primates. Family, Hominidae. Genus, Homo. Species, Homo sapiens. Example 2. Plant mango. Kingdom, Plantae. Division, Angiospermae. Class, Dicotyledonnae. Order, Sapindales. Family, Anacardiaceae. Genus, Mangifera. Species, Mangifera indica. This hierarchy shows how classification moves from broad categories kingdom to specific ones, species, representing an organism's exact position in the biological system. This is Biodecode, and you're listening to Chapter 2, Biological Classification. Chapter 2, Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific, but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, two-kingdom system of classification with Plantae and Animalia kingdoms was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two-kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics, like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. R.H. Whitaker, 1969, proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. Let's take a closer look at Table 2.1, which shows R.H. Whitaker's five-kingdom classification, posed in 1969. Whitaker classified all living organisms into five kingdoms: Monera, Protista, Fungi, Plantae, and Animalia. This classification was based on several key features, such as 1. Cell structure, whether organisms are prokaryotic or eukaryotic. 2. Body organization, whether they are unicellular or multicellular. 3. Mode of nutrition, whether they make their own food, autotrophic, or depend on others, heterotrophic. 4. Reproduction. 5. Phylogenetic relationships, which show their evolutionary connections. Now, let's understand the kingdoms one by one. As summarized in the table, Kingdom Monera includes all prokaryotic organisms, such as bacteria and cyanobacteria. They are unicellular, lack true nucleus, and reproduce asexually by binary fission. Kingdom Protista, made up of unicellular eukaryotes, such as amoeba, paramecium, and euglena. They have a true nucleus and show both autotrophic and heterotrophic modes of nutrition. Kingdom Fungi, mostly multicellular and eukaryotic, with a few unicellular forms like yeast. They are heterotrophic and saprophytic, meaning they feed on decaying organic matter. Kingdom Plantae, These are multicellular eukaryotes that perform photosynthesis. Their mode of nutrition is autotrophic, and they show a clear cell wall made of cellulose. Kingdom Animalia includes multicellular eukaryotic organisms that are heterotrophic, lack cell walls, and have well-developed systems for movement and response. This table helps us see at a glance how each kingdom differs in cell type, structure, nutrition, and organization. It laid the foundation for modern classification systems and helped biologists understand the diversity of life more scientifically. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationships. Table 2.1 gives a comparative account of different characteristics of the 5 kingdoms. The 3-domain system has also been proposed that divides the kingdom Monera into 2 domains, leaving the remaining eukaryotic kingdoms in the 3rd domain and thereby 6-kingdom classification. You will learn about the system in detail in higher classes. Let us look at this 5-kingdom classification to understand the issues and considerations that influence the classification system. Earlier classification systems included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperms under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organisms and the multicellular ones. Say, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group, fungi, and the autotrophic green plants, though they also showed a characteristic difference in their walls' composition. Fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, Kingdom Fungi. All prokaryotic organisms were grouped together under Kingdom Monera, and the unicellular eukaryotic organisms were placed in Kingdom Protista. Kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae within plants, and both having cell walls with paramecium and amoeba, which were earlier placed in the animal kingdom, which lack cell wall. It has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities, but is also phylogenetic. That is based on evolutionary relationships. In this chapter, we will study characteristics of kingdoms Monera, Protista, and Fungi of the Whittaker system of classification. Kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms, respectively, will be dealt separately in chapters 3 and 4. Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are the most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats, such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under 4 categories based on their shape: the spherical coccus pall, Toxi, the rod-shaped bacillus pall, Bacilli, the comma-shaped Vibrium pall, Vibrio, and the spiral Spirillum pall, Spirilla. Let's take a look at figure 2.1, which shows bacteria of different shapes. Bacteria are unicellular prokaryotes, meaning they lack a true nucleus. They come in a variety of shapes. Toxi, these are spherical bacteria, like tiny balls. They may occur singly, in pairs, chains, or clusters. Bacilli, these are rod-shaped bacteria, longer than they are wide. Spirilla, or spirochetes, these are spiral-shaped bacteria, which may be rigid or flexible. Observing their shapes, helps microbiologists identify and classify them, since the shape is often related to their habitat, movement, and how they interact with other organisms. Though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic. That is, they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs. That is, they depend on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats, such as extreme salty areas halophiles, hot springs, thermoacidophiles, and marshy areas methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals, such as cows and buffaloes, and they are responsible for the production of methane biogas from the dung of these animals. Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall, and if motile, a flagellum. Cyanobacteria, also referred to as blue-green algae, have chlorophyll, similar to green plants, and are photosynthetic autotrophs. Cyanobacteria are unicellular, colonial or filamentous, freshwater marine or terrestrial algae. Colonies are generally surrounded by a gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterocysts. For example, nostoc and Animalia. Chemosynthetic autotrophic bacteria oxidize various inorganic substances, such as nitrates, nitrites, ammonia, and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Next, we have figure 2.2, showing a filamentous blue-green algae called nostoc. Nostoc is a colonial cyanobacterium, forming long chains or filaments of cells enclosed in a gelatinous sheath. These organisms are photosynthetic, meaning they produce their own food using sunlight. They are also important in nitrogen fixation, helping enrich the soil with nitrogen, a nutrient essential for plants. By looking at this figure, you can clearly see how these cells connect in a chain-like structure, forming a visible filament under the microscope. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human 13 affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume roots, etc. Some are pathogens causing damage to human beings, crops, farm animals, and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission, figure 2.3. Sometimes, under unfavorable conditions, they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacterium to the other. Finally, let's look at figure 2.3, which shows a bacterium in the process of division. Bacteria reproduce asexually, primarily by binary fission, where one cell splits into two identical daughter cells. In the figure, you can see the cell elongating and the genetic material being copied. This simple yet efficient method allows bacteria to multiply rapidly under favorable conditions. Understanding this figure helps you visualize how bacterial populations grow and spread, which is important in both ecology and medicine. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be photosynthetic protistan to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom forms a link with the others dealing with plants, animals, and fungi. Being eukaryotes, Protista's cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Chrysophytes. This group includes diatoms and golden algae desmids. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water currents plankton. Most of them are photosynthetic. In diatoms, the cell walls form two thin overlapping shells, which fit together as in a soapbox. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind a large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the oceans. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red, depending on the main pigments present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often, red dinoflagellates. Example, gonulacs undergo such rapid multiplication that they make the sea appear red tides. Toxins released by such large numbers may even kill other marine animals, such as fishes. Euglenoids. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, pigments of euglenoids are identical to those present in higher plants. Example, Euglena figure 2.4b. Slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. Spores possess true walls. They are extremely resistant and survive for many years, even under adverse conditions. The spores are dispersed by air currents. Now, let's take a look at figure 2.4, which shows some common protists. Dinoflagellates are mostly marine plankton. They are unicellular and have two flagella that help them move. Some dinoflagellates are bioluminescent and can cause red tides in oceans. Euglena is a unicellular protist that shows both plant-like and animal-like features. It has chloroplasts so it can perform photosynthesis, but it can also take in food from the environment when light is absent. Slime molds are fungus-like protists. They can form multicellular aggregates and move like amoebae in their feeding stage. They are fascinating because they can behave like individual cells and also as a coordinated group. Paramecium is a well-known ciliate protist. It is covered with tiny hair-like structures called cilia, which help it move and sweep food particles into its oral groove. Observing these figures helps you understand the incredible diversity within Protista, from photosynthetic forms to mobile heterotrophs. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans. These organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia false feet, as in amoeba. Marine forms have silica shells on their surface. Some of them, such as entamoeba, are parasites. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic form cause diaceasis such as sleeping sickness. Example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be steered into the gullet. Example, Paramecium, figure 2.4. Sporozoans. This includes diverse organisms that have an infectious spore, like stage in their life cycle. The most notorious is Plasmodium malarial parasite, which causes malaria, a disease which has a staggering effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves are due to a parasitic fungus. Some unicellular fungi, for example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the source of antibiotics. For example, penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of yeasts, which are unicellular, fungi are filamentous. Their bodies consist of long, slender thread-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called coenocytic hyphae. Others have septae or cross walls in their hyphae. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates, and hence are called saprophytes. Those that depend on living plants and animals are called parasites. They can also live as symbionts in association with algae as lichens, and with roots of higher plants as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, and budding. Asexual reproduction is by spore called conidia, or sporangiospores, or zoospores, and sexual reproduction is by oospores, ascospores, and basidiospores. The various spores are produced in distinct structures called brooding bodies. The sexual cycle involves the following 3 steps. Step 1. Fusion of protoplasms between 2 motile or non-motile gametes called plasmogamy. Step 2. Fusion of 2 nuclei called karyogamy. Step 3. Meiosis in zygote, resulting in haploid spores. When a fungus reproduces sexually, 2 haploid hyphae of compatible mating types come together and fuse. In some fungi, the fusion of 2 haploid cells immediately results in diploid cells twin. However, in other fungi, ascomycetes and basidiomycetes, an intervening dikaryotic stage N+N, that is, 2 nuclei per cell, occurs. Such a condition is called a dikaryon, and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse, and the cells become diploid. The fungi form fruiting bodies in which reduction division occurred, leading to formation of haploid spores. The morphology of the mycelium, mode of spore formation and fruiting bodies form the basis for the division of the kingdom into various classes. Next, we have figure 2.5, showing examples of fungi. Mucor is a filamentous fungus commonly found on bread. It grows as a network of hyphae and reproduces by forming sporangiospores. Aspergillus is another filamentous fungus, often used in biotechnology. It reproduces both sexually and asexually, and has economic importance in enzyme production. Agaricus is the common mushroom, a multicellular fungus with a distinct fruiting body. It reproduces via spores and is widely consumed as food. These figures illustrate the different forms of fungi, from simple filamentous types to large fruiting bodies, highlighting their structural diversity and ecological role. Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. The mycelium is aseptate and coenocytic. Asexual reproduction takes place by zoospores, motile, or by aplanospores, non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous, or dissimilar, anisogamous or oogamous. Some common examples are Mucor figure 2.5a, Rhizopus the bread mold mentioned earlier, and Albugo, parasitic fungi on mustard. Ascomycetes. Commonly known as sac fungi, the ascomycetes are mostly multicellular. For example, penicillium, or really unicellular, for example, yeast Saccharomyces. They are saprophytic, decomposers, parasitic, or coprophilous, growing on dung. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on the special mycelium called conidiophores. Conidia on germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac like asci singular ascus. These asci are arranged in different types of fruiting bodies called ascocarps. Some examples are Aspergillus figure 2.5b, claviceps, and Neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morels and truffles are edible and are considered delicacies. Basidiomycetes. Commonly known forms of Basidiomycetes are mushrooms, bracket fungi, or puffballs. They grow in soil, on logs and tree stumps, and in living plant bodies as parasites, or exemplarusts and smuts. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strains or genotypes. The resultant structure is eukaryotic, which ultimately gives rise to basidium. Karyogamy and meiosis take place in the basidium, producing 4 basidiospores. Basidiospores are exogenously produced on the basidium pulp. Basidia. The basidia are arranged in fruiting bodies called basidiocarps. Some common members are agaricus mushroom, figure 2.5c, Eustalago smut, and Pocinia rustfungus. Deuteromycetes. Commonly known as imperfect fungi, because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered, they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of Deuteromycetes. Once perfect sexual stages of members of Deuteromycetes were discovered, they were often moved to Ascomycetes and Basidiomycetes. Deuteromycetes reproduce only by asexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are Alternaria, Colletotricum, and Trichoderma. Kingdom Plantae. Kingdom Plantae includes all eukaryotic chlorophyll containing organisms commonly called plants. A few members are partially heterotrophic, such as the insectivorous plants or parasites. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscuta is a parasite. Plant cells have an eukaryotic structure with prominent chloroplasts and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in Chapter 8. Plantae includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plants has 2 distinct phases. the diploid sporophytic and the haploid gametophytic, that alternate with each other. The length of the haploid and diploid phases, and whether these phases are free-living or dependent on others, vary among different groups in plants. This phenomenon is called alternation of generations. You will study further details of this kingdom in Chapter 3. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell walls. They directly or indirectly depend on plants for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic, by ingestion of food. They follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanism. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female, followed by embryological development. Salient features of various phyla are described in Chapter 4. Viruses, viroids, prions, and lichens. In the five-kingdom classification of Whitaker, there is no mention of lichens, and some acellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effects viruses can have on us, even if we do not associate it with our condition. Viruses did not find a place in classification since they are not considered truly living, if we understand living as those organisms that have a cell structure. The viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves, killing the host. Would you call viruses living or non-living? Finally, let's look at Figure 2.6, which shows some viruses. Tobacco mosaic virus TMV is a plant virus that infects tobacco and other plants. It has a rod-like structure and contains RNA as its genetic material. Bacteriophage is a virus that infects bacteria. It has a complex structure with a head that contains DNA, a tail, and tail fibers that help it attach to the bacterial cell. These figures help visualize the unique forms of viruses and remind us that, although cellular, viruses play a crucial role in both health and disease. Virus means venom or poisonous fluid. Dmitry Ivanovsky, 1892, recognized certain microbes as causal organism of the mosaic disease of tobacco. Figure 2.6a. These were found to be smaller than bacteria because they passed through bacteria-proof filters. M. W. Beijerinck, 1898, demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and named the new pathogen virus and called the fluid as contagium, vivum, fluidum, infectious living fluid. W. M. Stanley, 19, fluidum, infectious living fluid. W. M. Stanley, 1935, showed that viruses could be crystallized and crystals consist largely of proteins. They are inert outside their specific host cell. Viruses are obligate parasites. In addition to proteins, viruses also contain genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein, and the genetic material is infectious. In general, viruses that infect plants have single-stranded RNA, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. Bacterial viruses or bacteriophages viruses that infect the bacteria are usually double-stranded DNA viruses. Figure 2.6b. The protein coat called capsid, made of small subunits called capsomeres, protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause diseases like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing and stunted growth. Viroids. In 1971, T.O. Diener discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein coat that is found in viruses, hence the name viroid. The RNA of the viroid was of low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy (BSE), commonly called mad cow disease in cattle, and its analogous variant Creutzfeldt-Jakob disease (CJD). Lichens. Lichens are symbiotic associations, i.e. mutually useful associations, between algae and fungi. The algal component is known as phycobiont and fungal component as mycobiont, which are autotrophic and heterotrophic, respectively. Algae pair food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicators. They do not grow in polluted areas. Summary. Biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological characters. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Wicker proposed an elaborate five-kingdom classification, Monera, Protista, Fungi, Plantae, and Animalia. The main criteria of the five-kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In the five-kingdom classification, bacteria are included in Kingdom Monera. Bacteria are cosmopolitan in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista includes all single-celled eukaryotes, such as chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Protists have defined nucleus and other membrane-bound organelles. They reproduce both asexually and sexually. Members of Kingdom Fungi show a great diversity in structures and habitat. Most fungi are saprophytic in their mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes are the four classes under this kingdom. Plantae includes all eukaryotic chlorophyll-containing organisms. Algae, bryophytes, pteridophytes, gymnosperms, and angiosperms are included in this group. The life cycle of plants exhibit alternation of generations, gametophytic and sporophytic generations. The heterotrophic eukaryotic, multicellular organisms lacking a cell wall are included in the kingdom Animalia. You've just finished listening to Chapter 2, Biological Classification from NCERT Class 11 Biology, here on Biodecode. This is Biodecode, and you're listening to Chapter 2, Biological Classification, Exercise Solutions, and Key Questions. Question 1. Discuss how classification systems have undergone several changes over a period of time. Answer. The classification of living organisms has evolved continuously as our understanding of biology has deepened. Initially, Aristotle proposed a two-kingdom system that included only plants and animals. Later, with the invention of the microscope and discovery of microorganisms, the need for a broader system arose. In 1866, Ernst Haeckel added a third kingdom, Protista, to include unicellular organisms. Then, R.H. Whitaker in 1969 proposed the five-kingdom classification, Monera, Protista, Fungi, Plantae, and Animalia. Further research in molecular biology and genetics led to the modern three-domain system by Carl Woese, which divides life into bacteria, archaea, and eukarya. Thus, classification systems have changed from morphological to molecular approaches, reflecting advancements in science and technology. Question 2. State 2 economically important uses of a heterotrophic bacteria and B. Archaebacteria. Answer. Heterotrophic bacteria. 1. They play a key role in sewage treatment by decomposing organic waste. 2. Certain species, like lactobacillus, are used in the production of curd and cheese through fermentation. B. Archaebacteria. 1. Methanogens are used in biogas production from cow dung and other organic waste. 2. They also play a role in wastewater treatment by breaking down organic matter under anaerobic conditions. Question 3. What is the nature of cell walls in diatoms? Answer. Diatoms have cell walls made of silica, forming two overlapping halves called frustules. These walls are extremely hard and resistant to decay, giving rise to fossilized deposits known as diatomaceous earth. Question 4. Find out what do the terms algal bloom and red tide signify? Answer. An algal bloom refers to the rapid increase in the population of algae in water bodies, often due to nutrient enrichment. This can reduce oxygen levels and harm aquatic life. Red tides are a special type of algal bloom caused by certain marine dinoflagellates, such as goniolacs, which release toxins and turn the water reddish-brown. Question 5. How are viroids different from viruses? Answer. Viroids are smaller and simpler than viruses. They consist of a short-strand circular RNA without any protein coat, whereas viruses have both nucleic acid DNA or RNA and a protein capsid. Viroids infect only plants, while viruses infect both plants and animals. Question 6. Describe briefly the four major groups of protozoa. Answer. Protozoa are classified into four major groups based on locomotion. 1. Amoeboid protozoa move using pseudopodia, for example, amoeba. 2. Flagellated protozoa move with flagella, for example, anosoma. 3. Ciliated protozoa use cilia for movement, for example, paramecium. 4. Sporozoan, non-motile, parasitic forms, for example, Plasmodium, the malaria parasite. Question 7. Plants are autotrophic. Can you think of some plants that are partially heterotrophic? Answer. Yes, certain plants are partially heterotrophic because they obtain nutrients from other organisms. Examples include pitcher plant Nepenthes, Venus flytrap Dionea, sundew Drosera. These are insectivorous plants that trap and digest insects to fulfill their nitrogen requirement. Question 8. What do the terms icobiont and mycobiont signify? Answer. In lichens, the two partners are icobiont, which is the algal component that performs photosynthesis, and mycobiont, which is the fungal component that provides structure and protection. Together, they live in a symbiotic relationship, mutually benefiting each other. Question 9. Give a comparative account of the classes of kingdom fungi under the following. First, mode of nutrition, and second, mode of reproduction. Answer. First, mode of nutrition. All fungi are heterotrophic, obtaining food by absorption. They can be saprophytic, parasitic, or symbiotic. Second, mode of reproduction. Phycomycetes reproduce asexually by zoospores, sexually by zygospores. Ascomycetes reproduce asexually by conidia, sexually by ascospores within asci. Basidiomycetes, asexual spores are rare, sexually reproduce by basidiospores. Deuteromycetes reproduce only asexually by conidia. Their sexual stage is unknown. Question 10. What are the characteristic features of euglenoids? Answer. Euglenoids, such as Euglena, are unicellular organisms found in fresh water. They have a protein-rich pellicle instead of a cell wall, providing flexibility. They possess two flagella for movement and contain chloroplasts with chlorophyll for photosynthesis. In the absence of light, they can act as heterotrophs, showing both plant-like and animal-like features. Question 11. Give a brief account of viruses with respect to their structure and nature of genetic material. Also name 4 common viral diseases. Answer. Viruses are non-cellular infectious agents composed of nucleic acid, either DNA or RNA, enclosed in a protein coat called capsid. They lack cellular machinery and reproduce only inside a host cell. Examples of viral diseases include influenza, polio, AIDS, and COVID-19. Question 12. Organize a discussion in your class on the topic, are viruses living or non-living? Suggested discussion summary. Viruses exhibit both living and non-living characteristics. They are non-living outside a host, as they cannot reproduce or carry out metabolism. However, once inside a host cell, they become active and replicate, showing living traits. Hence, viruses are considered the borderline of living and non-living entities. This is Biodecode, and you're listening Chapter 3, Plant Kingdom. Chapter 3, Plant Kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whitaker in 1969, wherein he suggested the 5-kingdom classification, viz., Monera, Protista, Fungi, Animalia, and Plantae. In this chapter, we will deal in detail with further classification within Kingdom Plantae, popularly known as the plant kingdom. We must stress here that our understanding of the plant kingdom has changed over time. Fungi and members of the Monera and Protista having cell walls have now been excluded from Plantae, though earlier classifications placed them in the same kingdom. So, the cyanobacteria that are also referred to as blue-green algae are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under Plantae. Let us also look at classification within angiosperms to understand some of the concerns that influence the classification systems. The earliest systems of classification used only gross superficial morphological characters such as habit, color, number and shape of leaves, etc. They were based mainly on vegetative characters or on the Androecium structure system given by Linnaeus. Such systems were artificial. They separated the closely related species since they were based on a few characteristics. Also, the artificial systems gave equal weightage to vegetative and sexual characteristics. This is not acceptable, since we know that often the vegetative characters are more easily affected by environment. As against this, natural classification systems developed, which were based on natural affinities among the organisms and consider not only the external features, but also internal features, like ultrastructure, anatomy, embryology, and phytochemistry. Such a classification for flowering plants was given by George Bentham and Joseph Dalton Hooker. At present, phylogenetic classification systems based on evolutionary relationships between the various organisms are acceptable. This assumes that organisms belonging to the same taxa have a common ancestor. We now use information from many other sources too to help resolve difficulties in classification. These become more important when there is no supporting fossil evidence. Numerical taxonomy, which is now easily carried out using computers, is based on all observable characteristics. Number and codes are assigned to all the characters, and the data are then processed. In this way, each character is given equal importance, and at the same time, hundreds of characters can be considered. Cytotaxonomy, that is based on cytological information like chromosome number, structure, behavior, and chemotaxonomy, that uses the chemical constituents of the plant to resolve confusions, are also used by taxonomists these days. Algae. Algae are chlorophyll-bearing, simple, thalloid, autotrophic, and largely aquatic, both freshwater and marine organisms. They occur in a variety of other habitats, moist stones, soils, and wood. Some of them also occur in association with fungi, lichen, and animals, for example on sloth bear. The form and size of algae is highly variable, ranging from colonial forms like volvox and the filamentous forms like Ulothrix and Spirogyra. Figure 3.1. A few of the marine forms, such as kelps, form massive plant bodies. The algae reproduce by vegetative, asexual, and sexual methods. Vegetative reproduction is by fragmentation. Each fragment develops into a thallus. Asexual reproduction is by the production of different types of spores, the most common being the zoospores. They are flagellated motile, and on germination gives rise to new plants. Sexual reproduction takes place through fusion of two gametes. These gametes can be flagellated and similar in size as in Ulothrix, or non-flagellated, non-motile, but similar in size as in Spirogyra. Such reproduction is called isogamous. Fusion of two gametes dissimilar in size, as in species of Euderina, is termed as anisogamous. Fusion between one large, non-motile, static, female gamete and a smaller, motile, male gamete is termed oogamous. For example, volvox, fucus. Figure 3.1 shows representative examples of algae, divided into three groups: green algae, brown algae, and red algae. Let us go through each. Green algae, chlorophyceae. First, volvox, a colonial alga. It appears as a spherical colony of many cells arranged on the periphery of a gelatinous matrix. Each cell has flagella, and together they coordinate movement. Second, Ulothrix, filamentous form. Here, cells form a uniseriate filament, a single row of cells. Each cell contains chloroplasts, and there is no differentiation into roots or stems. Brown algae Phaeophyceae. First, Laminaria, often referred to as kelp. It has a differentiated body with a holdfast attachment organ, stipe stem-like, and a blade leaf-like. Second, Fucus, typically marine seaweed. You see a thallus, which may bear conceptacles, reproductive cavities, and often a midrib. Red algae, Rhodophyceae. First, Porphyra, a thin sheet-like alga also known as nori in culinary use. Second, Polysiphonia, a filamentous branched alga with several parallel filaments, siphons. And finally, Dictyota, a foliose leaf-like thalloid alga with dichotomous branching. This figure, therefore, illustrates the morphological diversity of algae, from unicellular, colonial, filamentous, to sheet-like and complex thalli. Use this to explain that algae is not a homogeneous group, but contains a variety of body forms. Algae are useful to man in a variety of ways. At least a half of the total carbon dioxide fixation on Earth is carried out by algae through photosynthesis. Being photosynthetic, they increase the level of dissolved oxygen in their immediate environment. They are of paramount importance as primary producers of energy-rich compounds, which form the basis of the food cycles of all aquatic animals. Many species of Porphyra, Laminaria, and Sargassum are among the 70 species of marine algae used as food. Certain marine brown and red algae produce large amounts of hydrocolloids, water-holding substances. For example, algin brown algae and carrageen red algae, which are used commercially. Agar, one of the commercial products obtained from Gelidium and Gracilaria, are used to grow microbes and in preparations of ice creams and jellies. Chlorella, a unicellular alga rich in proteins, is used as food supplement even by space travelers. The algae are divided into three main classes: Chlorophyceae, Phaeophyceae, and Rhodophyceae. Chlorophyceae. The members of Chlorophyceae are commonly called green algae. The plant body may be unicellular, colonial, or filamentous. They are usually grass-green due to the dominance of pigments chlorophyll A and B. The pigments are localized in definite chloroplasts. The chloroplasts may be discoid, plate-like, reticulate, cup-shaped, spiral, or ribbon-shaped in different species. Most of members have one or more storage bodies called pyrenoids, located in the chloroplasts. Pyrenoids contain protein besides starch. Some algae may store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner layer of cellulose and an outer layer of pectose. Vegetative reproduction usually takes place by fragmentation. Asexual reproduction is by flagellated zoospores produced in zoosporangia. The sexual reproduction shows considerable variation in the type and formation of sex cells, and it may be isogamous, anisogamy, or oogamous. Some commonly found green algae are Chlamydomonas, Volvox, Ulothrix, Spirogyra, and Chara, figure 3.0. Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily in marine habitats. They show great variation in size and form. They range from simple branched, filamentous forms ectocarpus to profusely branched forms as represented by kelps, which may reach a height of 100 meters. They possess chlorophyll A, C, carotenoids, and xanthophylls. They vary in color from olive green to various shades of brown, depending upon the amount of the xanthophyll pigment, fucoxanthin, present in them. Food is stored as complex carbohydrates, which may be in the form of laminarin or mannitol. The vegetative cells have a cellulosic wall usually covered on the outside by a gelatinous coating of algin. The protoplast contains, in addition to plastids, a centrally located vacuole and nucleus. The plant body is usually attached to the substratum by a holdfast and has a stalk, stipe, and leaf-like photosynthetic organ, the frond. Vegetative reproduction takes place by fragmentation. Asexual reproduction in most brown algae is by biflagellate zoospores that are pear-shaped and have two unequal laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Union of gametes may take place in water or within the Oogonium oogamous species. The gametes are piriform pear-shaped and bear two laterally attached flagella. The common forms are ectocarpus, dictyota, laminaria, sargassum, and fucus, figure 3.1. Rhodophyceae. The members of Rhodophyceae are commonly called red algae because of the predominance of the red pigment, R-phycoerythrin, in their body. Majority of the red algae are marine with greater concentrations found in the warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in oceans where relatively little light penetrates. The red thalli of most of the red algae are multicellular. Some of them have complex body organization. The food is stored as fluoridine starch, which is very similar to amylopectin and glycogen in structure. Let's now move to Table 3.1 in your NCERT textbook. This table summarizes the three major divisions of algae and compares their main distinguishing features, including pigments, stored food, cell wall composition, habitat, and reproduction. Let's go through each division one by one so that you can clearly visualize the differences. First one is green algae, Chlorophyceae. First, we have the green algae, scientifically known as Chlorophyceae. As the name suggests, their green color is due to the dominance of chlorophyll A and chlorophyll B pigments. They store their reserve food material in the form of starch, very similar to higher plants. The cell wall is made up of cellulose, giving it a firm structure. Green algae are mostly found in freshwater habitats, such as ponds, ditches, and moist soil, although some species also occur in marine environments. They reproduce by all three methods: vegetative reproduction by fragmentation, asexual reproduction through motile zoospores, and sexual reproduction, which may show isogamy, anisogamy, or oogamy, depending on the species. Examples include Chlamydomonas, Volvox, Ulothrix, Spirogyra, and Chara. So whenever you see bright green algae in a pond, it's most likely from this group. Second, brown algae, Phaeophyceae. Next, we move to the brown algae, or Phaeophyceae. Their characteristic brown or olive green color is due to the presence of the pigment fucoxanthin, along with chlorophyll A and C. They store food in the form of laminarin or mannitol instead of starch. Their cell wall has two layers, the inner layer made of cellulose and the outer layer composed of algin, a gelatinous substance. Brown algae are predominantly marine, found attached to rocks in coastal regions and cold seas. They exhibit great variation in size and form, from simple filaments to giant kelps that can reach several meters in length. Reproduction occurs by vegetative, asexual, and sexual methods. Sexual reproduction is mostly oogamous, and the gametes are often motile. Typical examples are Ectocarpus, Dictyota, Laminaria, Sargassum, and Fucus. These are the large seaweeds you often see washed up on seashores. Third, red algae, Rhodophyceae. Finally, we have the red algae, called Rhodophyceae. Their distinctive red color comes from the accessory pigment R-phycoerythrin, which masks the green of chlorophyll A and D. This pigment allows them to absorb blue light and thus thrive in deeper marine water where other wavelengths cannot penetrate. Their stored food is mainly Floridian starch, and the cell wall is composed of cellulose and polysulfate esters like carrageenan or agar. Most red algae are marine and prefer warm waters, although a few can be found in freshwater. Asexual reproduction takes place through non-motile spores called monospores, and sexual reproduction is oogamous. Notably, male and female gametes are non-motile, an important characteristic of this group. Common examples include Polysiphonia, Porphyra, Gracilaria, and Gelidium, some of which are used commercially for extracting agar and carrageenan. The red algae usually reproduce vegetatively by fragmentation. They reproduce asexually by non-motile spores and sexually by non-motile gametes. Sexual reproduction is oogamous and accompanied by complex post-fertilization developments. The common numbers are Polysiphonia, Porphyra figure 3.1ec, Gracilaria, and Gelidium. Bryophytes. Bryophytes include the various mosses and liverworts that are found commonly growing in moist shaded areas in the hills. Figure 3.2. Figure 3.2 demonstrates bryophytes, specifically the liverwort Marchantia and mosses, e.g. Funaria, sphagnum. The female thallus of Marchantia shows lobed, flat, prostrate structure. You might see archegoniophores, female reproductive stalks, arising from the thallus. Female thallus of Marchantia. Similar thalloid body, but with antherideophores, male reproductive stalks bearing antheridia on top. These two images help you to distinguish male and female gametophytes in liverworts. See Funaria moss, combined gametophyte and sporophyte. The leafy green gametophyte supports a slender sporophyte with seta stalk and capsule sporangium at its apex. The sphagnum gametophyte shows the dense carpet of moss, often with many slender photosynthetic branches, and sometimes red or green coloration. Sphagnum lacks a conspicuous sporophyte in this image. Focus is on the gametophyte. This figure emphasizes that bryophytes have a dominant gametophyte generation, and their sporophyte depends on the gametophyte. Bryophytes are also called amphibians of the plant kingdom, because these plants can live in soil but are dependent on water for sexual reproduction. They usually occur in damp, humid, and shaded localities. They play an important role in plant succession on bare rock soil. Plant body of bryophytes is more differentiated than that of algae. It is thallus-like and prostrate or erect, and attached to the substratum by unicellular or multicellular rhizoids. They lack true roots, stem, or leaves. They may possess root-like, leaf-like, or stem-like structures. The main plant body of the bryophyte is haploid. It produces gametes, hence is called a gametophyte. The sex organs in bryophytes are multicellular. The male sex organ is called antheridium. They produce biflagellate antherozoids. The female sex organ, called archegonium, is flask-shaped and produces a single egg. The antherozoids are released into water where they come in contact with archegonium. An antherozoid fuses with the egg to produce the zygote. Zygotes do not undergo reduction division immediately. They produce a multicellular body called a sporophyte. The sporophyte is not free-living, but attached to the photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction division meiosis to produce haploid spores. These spores germinate to produce gametophyte. Bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, birds, and other animals. Species of sphagnum, a moss, provide peat that have long been used as fuel and as packing material for transshipment of living material because of their capacity to hold water. Mosses, along with lichens, are the first organisms to colonize rocks and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. The bryophytes are divided into liverworts and mosses. Liverworts. The liverworts grow usually in moist, shady habitats, such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. The plant body of a liverwort is thalloid, for example Martantia. The thallus is dorsiventral and closely oppressed to the substrate. The leafy members have tiny leaf-like appendages in 2 rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli or by the formation of specialized structures called gemmae. Gemma are green, multicellular, asexual buds which develop in small receptacles called gemma cups located on the thalli. Gemmae become detached from the parent body and germinate to form new individuals. During sexual reproduction, male and female sex organs are produced either on the same or on different thalli. The sporophyte is differentiated into a foot, seta, and capsule. After meiosis, spores are produced within the capsule. These spores germinate to form free-living gametophytes. Mosses. The predominant stage of the life cycle of a moss is the gametophyte, which consists of 2 stages. The first stage is the protonema stage, which develops directly from a spore. It is a creeping green, branched, and frequently filamentous stage. The second stage is the leafy stage, which develops from the secondary protonema as a lateral bud. It consists of upright, slender axes bearing spirally arranged leaves. They are attached to the soil through multicellular and branched rhizoids. This stage bears the sex organs. Vegetative reproduction in mosses is by fragmentation and budding in the secondary protonema. In sexual reproduction, the sex organs antheridia and archegonia are produced at the apex of the leafy shoots. After fertilization, the zygote develops into a sporophyte consisting of a foot, seta, and capsule. The sporophyte in mosses is more elaborate than that in liverworts. Capsule contains spores. Spores are formed after meiosis. The mosses have an elaborate mechanism of spore dispersal. Common examples of mosses are Funaria, Polytrichum, and Sphagnum, figure 3.2. Pteridophytes. The pteridophytes include horsetails and ferns. Pteridophytes are used for medicinal purposes and as soil binders. They are also frequently grown as ornamentals. Evolutionarily, they are the first terrestrial plants to possess vascular tissues, xylem and phloem. You shall study more about these tissues in Chapter 6. The pteridophytes are found in cool, damp, shady places, though some may flourish well in sandy soil conditions. You may recall that in bryophytes, the dominant phase in the life cycle is the gametophytic plant body. However, in pteridophytes, the main plant body is a sporophyte, which is differentiated into true root, stem, and leaves. Figure 3.3. These organs possess well-differentiated vascular tissues. Leaves in Pteridophyta are small microphyls, as in Selagella, or large macrophyls, as in ferns. Sporophytes bear sporangia that are subtended by leaf-like appendages called sporophylls. In some cases, sporophylls may form distinct compact structures called strobili or cones, Selagella, equisetum. The sporangia produce spores by meiosis in spore mother cells. The spores germinate to give rise to inconspicuous, small but multicellular. Figure 3.3. Pteridophytes. In Figure 3.3, we see typical representatives of pteridophytes. Vascular cryptogams, Selagella, one of the lycophytes, with small scale-like leaves microphyls, shows branching and perhaps strobili spore cone. B. Equisetum, commonly called horsetail. Stems are jointed, hollow, with whorls of small reduced leaves. C. Fern, a classical fern frond is shown, typically with pinnate leaves divided segments. You may also notice the rhizome and roots. D. Salvinia, an aquatic heterosporous fern, floating leaves, submerged leaves, and rootlets may be shown. Here, the figure highlights that pteridophytes have true vascular tissues, xylem and phloem, have distinct roots, stems, leaves, and that they reproduce by spores, not seeds. Also, Salvinia is a useful example to introduce heterospory microspores and megaspores. Free-living, mostly photosynthetic thalloid gametophytes called prothallus. These gametophytes require cool, damp, shady places to grow. Because of this specific restricted requirement and the need for water for fertilization, the spread of living pteridophytes is limited and restricted to narrow geographical regions. The gametophytes bear male and female sex organs called antheridia and archegonia, respectively. Water is required for transfer of antherizoids, the male gametes released from the antheridia, to the mouth of archegonium. Fusion of male gamete with the egg present in the archegonium result in the formation of zygote. Zygote thereafter produces a multicellular, well-differentiated sporophyte, which is the dominant phase of the pteridophytes. In the majority of the pteridophytes, all the spores are of similar kinds. Such plants are called homosporous. Genera like Selagella and Salvinia, which produce two kinds of spores, macro, large, and micro, small spores, are known as heterosporous. The megaspores and microspores germinate and give rise to female and male gametophytes, respectively. The female gametophytes in these plants are retained on the parent sporophytes for variable periods. Development of the zygotes into young embryos take place within the female gametophytes. This event is a precursor to the seed habit, considered an important step in evolution. The pteridophytes are further classified into four classes: Psilopsida, Psilotum, Lycopsida, Selagella, Lycopodium, Sphenopsida, Equisetum, and Paropsida Triopteris, Paris adiantum. Gymnosperms. The gymnosperms. Gymnos, naked, sperma, seeds, are plants in which the ovules are not enclosed by any ovary wall and remain exposed, both before and after fertilization. The seeds that develop post-fertilization are not covered, that is, are naked. Gymnosperms include medium-sized trees or tall trees and shrubs. Figure 3.4. One of the gymnosperms, the giant red tree sequoia, is one of the tallest tree species. The roots are generally taproots. Roots in some genera have fungal association in the form of mycorrhiza pinus, while in some others, cycas, small specialized roots called coralloid roots, are associated with N2, fixing cyanobacteria. The stems are unbranched, associated with N2, fixing cyanobacteria. The stems are unbranched cycas or branched pinus, cedrus. The leaves may be simple or compound. In cycas, the pinnate leaves persist for a few years. The leaves in gymnosperms are well adapted to withstand extremes of temperature, humidity, and wind. In conifers, the needle-like leaves reduce the surface area. Their thick cuticle and sunken stomata also help to reduce water loss. The gymnosperms are heterospores. They produce haploid microspores and megaspores. The two kinds of spores are produced within sporangia that are borne on sporophylls, which are arranged spirally along an axis to form LAX, or compact strobili, or cones. The strobili-bearing or cones. The strobili-bearing microsporophylls and microsporangia are called microsporangiate or male strobili. The microspores develop into a male gametophytic generation, which is highly reduced and is confined to only a limited number of cells. This reduced gametophyte is called a pollen grain. The development of pollen grains take place within the microsporangia. The cones-bearing megasporophylls with ovules or megasporangia are called macrosporangiate or female strobili. The male or female cones or strobili may be borne on the same tree Pinus. However, in cycas, male cones and megasporophylls are borne on different trees. The megaspore mother cell is differentiated from one of the cells of the nucellus. The nucellus is protected by envelopes, and the composite structure is called an ovule. The ovules are borne on megasporophylls, which may be clustered to form the female cones. Megaspore mother cell divides meiotically to form 4 megaspores. One of the megaspores enclosed within the megasporangium develops into a multicellular female gametophyte that bears 2 or more archegonia or female sex organs. The multicellular female gametophyte is also retained within megasporangium. Unlike bryophytes and pteridophytes, in gymnosperms, the male and the female gametophytes do not have an independent free-living existence. They remain within the sporangia, retained on the sporophytes. The pollen grain is released from the microsporangium. They are carried in air currents and come in contact with the opening of the ovules borne on megasporophylls. The pollen tube carrying the male gametes grows towards archegonia in the ovules and discharge their contents near the mouth of the archegonia. Following fertilization, zygote develops into an embryo and the ovules into seeds. These seeds are not covered. Figure 3.4, Gymnosperms. Figure 3.4 presents gymnosperms, seed plants whose seeds are naked, not enclosed in an ovary. A cycas often shows a crown of pinnate leaves at the apex of a trunk, may also show male and female cones. B. pinus displays the familiar evergreen tree habit, needle leaves, and male-female cones. C. ginkgo, living fossil, with fan-shaped leaves, seeds borne on stalks, not in fruits. This figure helps you to contrast gymnosperms with earlier plant groups. Gymnosperms are vascular, have seeds, but do not form flowers or fruits. Note the structural adaptations to terrestrial life, woody tissue, persistent leaves, and reproductive cones. Angiosperms. Unlike the gymnosperms where the ovules are naked, in the angiosperms or flowering plants, pollen grains and ovules are developed in specialized structures called flowers. In angiosperms, the seeds are enclosed in fruits. The angiosperms are an exceptionally large group of plants occurring in wide range of habitats. They range in size from the smallest wolfia to tall trees of eucalyptus over 100 meters. They provide us with food, fodder, fuel, medicines, and several other commercially important products. They are divided into 2 classes, the dicotyledons and the monocotyledons, figure 3.5. Finally, figure 3.5 illustrates 2 types of flowering plants. A dicotyledon dicot. You may see a typical plant with 2 cotyledons in seedling, knit-like venation in leaves, flower parts in multiples of 4 or 5, and taproot system. A monocotyledon monocot shows 1 cotyledon in the seedling, parallel venation in leaves, flower parts in multiples of 3, root systems often fibrous, and stem cross-section structure. This figure is critical when teaching how monocots and dicots differ in seed structure, leaf venation, floral parts, root systems, and stem anatomy. Summary. Plant kingdom includes algae, bryophytes, peridophytes, gymnosperms, and angiosperms. Algae are chlorophyll-bearing simple, thalloid, autotrophic, and largely aquatic organisms. Depending on the type of pigment possessed and the type of stored food, algae are classified into three classes, namely Chlorophyceae, Aophyceae, and Rhodophyceae. Algae usually reproduce vegetatively by fragmentation, asexually by formation of different types of spores, and sexually by formation of gametes which may show isogamy, anisogamy, or oogamy. Bryophytes are plants which can live in soil but are dependent on water for sexual reproduction. Their plant body is more differentiated than that of algae. It is thallus-like and prostrate or erect and attached to the substratum by rhizoids. They possess root-like, leaf-like, and stem-like structures. The bryophytes are divided into liverworts and mosses. The plant body of liverworts is thalloid and dorsiventral, whereas mosses have upright, slender axes bearing spirally arranged leaves. The main plant body of a bryophyte is gamete-producing and is called a gametophyte. It bears the male sex organs called antheridia and female sex organs called archegonia. The male and female gametes produced fuse to form zygote, which produces a multicellular body called sporophyte. It produces haploid spores. The spores germinate to form gametophytes. In pteridophytes, the main plant is a sporophyte, which is differentiated into true root, stem, and leaves. These organs possess well-differentiated vascular tissues. The sporophytes bear sporangia which produce spores. The spores germinate to form gametophytes which require cool, damp places to grow. Gametophytes bear male and female sex organs, called antheridia and archegonia, respectively. Water is required for transfer of male gametes to archegonium where zygote is formed after fertilization. The zygote produces a sporophyte. The gymnosperms are the plants in which ovules are not enclosed by any ovary wall. After fertilization, the seeds remain exposed, and therefore these plants are called naked-seeded plants. The gymnosperms produce microspores and megaspores, which are produced in microsporangia and megasporangia borne on the sporophylls. The sporophylls, microsporophylls, and megasporophylls are arranged spirally on axis to form male and female cones respectively. The pollen grain germinates and pollen tube releases the male gamete into the ovule, where it fuses with the egg cell in archegonia. Following fertilization, the zygote develops into embryo and the ovules into seeds. The angiosperms are divided into two classes, the dicotyledons and the monocotyledons. You've just finished listening to Chapter 3, Plant Kingdom from NCERT, Class 11 Biology, here on Biodecode. This is Biodecode, and you're listening to Chapter 3, Plant Kingdom, Exercise Solutions and Key Questions. Question 1. What is the basis of classification of algae? Answer. The classification of algae is mainly based on three important features. First, the type of photosynthetic pigments they contain. Second, the nature of stored food material. And third, the composition of their cell wall. In Chlorophyceae, we find chlorophyll A and D, and they stored starch as reserve food. In Phaeophyceae, or brown algae, chlorophyll A, C, and phocoxanthin are present, giving them a brownish color, and they store laminarin and mannitol. Whereas Rhodophyceae, or red algae, have chlorophyll A and D, along with phycoerythrin, and store Floridian starch. So, in short, algae are classified based on pigment composition, storage products, and cell wall structure. Question 2. When and where does reduction division take place in the life cycle of a liverwort, a moss, a fern, a gymnosperm, and an angiosperm? Answer. Reduction division, also called meiosis, occurs when the diploid sporophyte produces haploid spores. Let's go group by group. In liverworts, meiosis takes place inside the capsule of the sporophyte during spore formation. In mosses, it occurs in the sporangium of the sporophyte. In ferns, meiosis happens within the sporangia borne on the sporophylls. In gymnosperms, meiosis takes place in the microsporangia and megasporangia of cones. And in angiosperms, it occurs in the anther and ovule, that is, in the microsporangium and megasporangium. So in all plant groups, reduction division takes place during spore formation in the sporophyte phase. Question 3. Name 3 groups of plants that bear archegonia. Briefly describe the life cycle of any one of them. Answer. The 3 groups of plants that possess archegonia are bryophytes, peridophytes, and gymnosperms. Let's take the example of a moss, bryophyte. In mosses, the dominant phase is the haploid gametophyte, which has 2 stages, protonoma stage and the leafy stage. Protona gives rise to the leafy gametophore that bears both male and female sex organs, the antheridia and archegonia. Fertilization takes place in the presence of water, forming a diploid zygote, which develops into a sporophyte. The sporophyte produces haploid spores by meiosis, and these spores germinate to form a new protonoma. Thus, the life cycle shows a clear alternation of generations between haploid and diploid phases. Question 4. Mention the ploidy of the following. Let's go one by one. Protonemal cell of a moss is haploid. Primary endosperm nucleus in dicot is triploid. Leaf cell of a moss is haploid. Prothallus cell of a fern is haploid. Gemma cell in Marchantia is haploid. Meristem cell of monocot is diploid. Ovum of a liverwort is haploid. Zygote of a fern is diploid. Question 5. Write a note on the economic importance of algae and gymnosperms. Answer. Let's begin with algae. Algae are economically important in several ways. They serve as food sources, as in Porphyra, Laminaria, and Sargassum. Industrially, they provide valuable substances like algin from brown algae and carrageenan from red algae. Blue-green algae such as Anabina and nostoc act as biofertilizers by fixing atmospheric nitrogen. They also play a major role in oxygen production and are now explored for biofuels and bioplastics. Now, gymnosperms also have great economic significance. They yield timber and softwood, pinus, cedrus, resins and gums like turpentine from pinus, and medicinal compounds like ephedrine from ephedra. They're also used as ornamental plants, for example, cycas and thuja, and provide pulpwood for the paper industry. Question 6. Both gymnosperms and angiosperms bear seeds. Then why are they classified separately? Answer. Although both groups produce seeds, they differ in how those seeds are enclosed and developed. In gymnosperms, the seeds are naked. They are not enclosed within fruits. Their ovules are exposed on the surface of megasporophylls, and they do not form flowers. In angiosperms, the seeds are enclosed within fruits. Ovules are protected inside the ovary, and true flowers are present. Moreover, the endosperm in gymnosperms is haploid, while in angiosperms it is triploid. Thus, gymnosperms are called naked seed plants, and angiosperms are enclosed seed plants, which justifies their separate classification. Question 7. What is heterospory? Briefly comment on its significance. 2 examples. Answer. Heterospory means the production of 2 different kinds of spores. Microspores, which form male gametophytes, and megaspores, which form female gametophytes. This condition is seen in Selagella and Salvinia. Its significance lies in evolution. It marks an important step toward the seed habit. The female gametophyte develops within the megaspore, remaining protected and nourished by the parent sporophyte. Hence, heterospory is a key evolutionary advancement leading to the origin of seeds. Question 8. Explain briefly the following terms with suitable examples. Answer. First, protonema. It is the first green filamented stage of the moss plant, developing from a spore. It produces buds that give rise to the leafy gametophyte. Example, Funaria. Second, antheridium. The male reproductive organ found in bryophytes, peridophytes, and gymnosperms. It produces motile male gametes called antherizoids. Third, artigonium. The female reproductive organ, flask-shaped, containing a single egg cell, present in bryophytes, peridophytes, and gymnosperms. Fourth, diplonic. A life cycle in which the diploid sporophyte is the dominant phase. Example, gymnosperms and angiosperms. Fifth, sporophyll. A leaf that bears sporangia, which produce spores. Example, the microsporophylls and megasporophylls of cycas. Sixth, isogamy. Form of sexual reproduction where gametes are similar in size and structure, though physiologically different. Example, Ulothrix and Sparagyra. Question 9. Differentiate between the following. Answer. First, red algae and brown algae. Red algae are mostly marine and found in deeper waters. They have chlorophyll A and D and phycoerythrin, the major pigment. They store food as scleroidean starch. Brown algae, on the other hand, are marine and found in colder seas. They have chlorophyll A, C, and phocoxanthin, and store mannitol and laminarin as reserve food. Second, liverworts and mosses. Liverworts have a thalloid plant body, while mosses have a leafy gametophyte. The liverwort sporophyte is short-lived, while in mosses it is more developed and has a capsule for spore formation. Liverworts reproduce by gemme, whereas mosses show a protonema stage. Third, homosporous and heterosporous pteridophytes. Homosporous pteridophytes produce only one type of spore, which gives rise to a bisexual gametophyte, for example, pteris. Heterosporous pteridophytes produce two kinds of spores, microspores and megaspores, which develop into unisexual gametophytes, for example, Selaginella. Question 10. Match the following. Chlamydomonas, algae. Cycas, gymnosperm. Selaginella, parrotphyte. Sphagnum, moss. Question 11. Describe the important characteristics of gymnosperms. Answer. Gymnosperms are mostly evergreen, woody trees or shrubs. They have a well-developed vascular system. Their xylem lacks vessels, and phloem lacks companion cells. They are heterosporous, producing microspores and megaspores. Their reproductive structures are organized into cones. Microsporangiate cones for male spores and megasporangiate cones for female spores. Seeds are naked, not enclosed within fruits. The gametophyte is highly reduced and dependent on the sporophyte. Pollination occurs through wind, and fertilization does not require water. The sporophyte is the dominant independent generation, showing a diplontic life cycle. Common examples include cycas, pinus, and ephedra. This is Biodecode, and you're listening to Chapter 4, Animal Kingdom. Chapter 4, Animal Kingdom. When you look around, you will observe different animals with different structures and forms. As over 1 million species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. Basis of classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, patterns of digestive, circulatory, or reproductive systems. These features are used as the basis of animal classification, and some of them are discussed here. Levels of organization. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregates, that is, they exhibit cellular level of organization. Some division of labor activities occur among the cells. In coelenterates, the arrangement of cells is more complex. Here, the cells performing the same function are arranged into tissues, hence is called tissue level of organization. A still higher level of organization, that is organ level, is exhibited by members of Platyhelminthes and other higher phyla where tissues are grouped together to form organs, each specialized for a particular function. In animals like annelids, arthropods, mollusks, echinoderms, and chordates, organs have associated to form functional systems, each system concerned with a specific physiological function. This pattern is called organ system level of organization. Organ systems in different groups of animals exhibit various patterns of complexities. For example, the digestive system in Platyhelminthe has only a single opening to the outside of the body that serves as both mouth and anus, and is hence called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory system may be of two types. First, open type in which the blood is pumped out of the heart, and the cells and tissues are directly bathed in it, and second, closed type in which the blood is circulated through a series of vessels of varying diameters, arteries, veins, and capillaries. Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical. That is, any plane that passes through the center does not divide them into equal halves. When any plane passing through the central axis of the body divides the organism into two identical halves, it is called radial symmetry. Coelenterates, ctenophores, and echinoderms have this kind of body plan, figure 4.1a. Animals like annelids, arthropods, and so on, where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. Figure 4.1b. Figure 4.1a. Radial symmetry. In this diagram, the organism's body is shown arranged around its central axis, like spokes radiating from the hub of a wheel. Free plane passing through the central axis divides the body into two identical halves. This type of symmetry is common among aquatic animals such as Hydra, Aurelia jellyfish, and starfish during certain life stages. Radial symmetry allows these organisms to interact with the environment from all sides equally, an adaptation suited for sessile non-moving or free-floating lifestyles. It ensures sensory structures and tentacles are evenly distributed around the body, which helps in food capture and defense, regardless of the direction of stimulus. Figure 4.1b, bilateral symmetry. Here, the figure demonstrates a body plan where only a single plane, called the median sagittal plane, can divide the organism into two identical halves, left and right. This symmetry is seen in higher animals such as annelids, arthropods, and vertebrates. Bilateral symmetry marks a major evolutionary advancement because it allows cephalization, or concentration of sense organs and nervous tissues at the anterior end, leading to directional movement and complex behavior. It signifies an active lifestyle where the animal moves in a definite direction and requires a coordinated body design. Diploblastic and triploblastic organization. Animals in which the cells are arranged in two embryonic layers, an external ectoderm and an internal endoderm, are called diploblastic animals. For example, coelenterids. An undifferentiated layer, mesoglia, is present in between the ectoderm and the endoderm, figure 4.2a. Those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and endoderm are called triploblastic animals, platyhelminthes, 2 chordates, figure 4.2b. Figure 4.2, germinal layers, a diploblastic, B, triploblastic. This figure compares the early embryonic organization of animals based on the number of primary germ layers formed during development. In the diploblastic structure A, the embryo has two layers, the ectoderm outer layer and endoderm inner layer, with a non-cellular jelly-like mesoglia in between. Examples include Hydra and Obelia. In contrast, the triploblastic structure B shows the presence of a third layer, the mesoderm, sandwiched between the ectoderm and endoderm. This additional layer leads to the formation of complex organs and organ systems. Animals like annelids, arthropods, mollusks, and chordates are triploblastic. The transition from diploblastic to triploblastic organization represents a key step in evolutionary complexity in body specialization. Palum. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is lined by mesoderm, is called coelom. Animals possessing coelom are called coelomates. For example, annelids, mollusks, arthropods, chynoderms, hemichordates, and chordates, figure 4.3a. In some animals, the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom, and the animals possessing them are called pseudocoelomates. For example, Aschelminthes, figure 4.3b. The animals in which the body cavity is absent are called acoelomates. For example, Platyhelminthes, figure 4.3c. Figure 4.3. Diagrammatic sectional view of body cavities. This comparative diagram illustrates three types of internal body cavity arrangements. Palomate, the true body cavity, coelom lies between the body wall and gut, lined by mesodermal tissue on both sides. It provides space for organ development, movement, and cushioning. Examples include annelids, mollusks, arthropods, and chordates. Pseudocoelomate, the body cavity is present but not completely lined by mesoderm. Instead, it lies between the mesoderm and endoderm. Found in roundworms ascaris, it allows limited organ movement and serves as a hydrostatic skeleton. The coelomate, no body cavity is present. Mesoderm fills the entire space between the body wall and gut. Flatworms planaria, Pania are typical examples. This figure highlights how the evolution of a coelom improved internal organization and organ efficiency. Segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation, and the phenomenon is known as metamerism. Notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates, and those animals which do not form this structure are called non-chordates. For example, porifera to echinoderms. Classification of animals. The broad classification of Animalia, based on common fundamental features as mentioned in the preceding sections, is given in Figure 4.4. The important characteristic features of the different phyla are described. Figure 4.4. Broad classification of kingdom Animalia, based on fundamental features. This figure presents a schematic tree showing how the vast diversity of animals can be systematically grouped based on key structural and functional traits. The classification starts from basic levels like cellular and tissue organization, body symmetry, and presence of a coelom, progressing to complex traits like segmentation and notochord development. The figure emphasizes the evolutionary transition from simple asymmetrical sponges porifera to more complex bilaterally symmetrical coelomate chordates. Each branching point represents the emergence of a new structural innovation that defines a distinct phylum. This evolutionary roadmap helps students visualize how animal complexity evolved step by step over time. Phylum Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetrical animals. Figure 4.5. These are primitive multicellular animals and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute pores ostia in the body wall into a central cavity, spongiocule, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange, and removal of waste. Choanocytes or collar cells line the spongiocule and the canals. Digestion is intracellular. Body is supported by a skeleton made up of spicules or spongin fibers. Sexes are not separate hermaphrodite. That is, eggs and sperms are produced by the same individual. Sponges reproduce asexually by fragmentation, and sexually by formation of gametes. Fertilization is internal, and development is indirect, having a larval stage which is morphologically distinct from the adult. Figure 4.5. Examples of porifera. Dicycon de Euthpongia C. Spongilla. This figure displays the characteristic body forms of sponges, organisms belonging to the phylum Porifera, the simplest multicellular animal. Cycon cypha has a vase-shaped body with numerous pores called ostia on its outer wall, leading to internal canals. The canal system filters water and extracts food particles, demonstrating the sponge's unique filter-feeding mechanism. Euthpongia bath sponge possesses a soft, fibrous skeleton made of spongin fibers, which allows flexibility. It represents the commercial sponges used for bathing. Spongilla, a freshwater sponge, unlike most marine species, forming greenish colonies due to symbiotic algae. This figure highlights the transition from simple cell aggregates to organisms with functional coordination via a canal system, an evolutionary step toward tissue-level organization. Examples: Cycon cypha, Spongilla, freshwater sponge, and Euthpongia bath sponge. Phylum Oenterata cnidaria. They are aquatic, mostly marine, sessile or free-swimming, radially symmetrical animals. Figure 4.6. The name cnidaria is derived from the cnidoblasts or cnidocytes, which contain the stinging capsules or nematocysts present on the tentacles and the body. Cnidoblasts are used for anchorage, defense, and for the capture of prey. Figure 4.7. Cnidarians exhibit tissue level of organization and are diploblastic. They have a central gastrovascular cavity with a single opening, mouth on hypostome. Digestion is extracellular and intracellular. Some of the cnidarians, for example corals, have a skeleton composed of calcium carbonate. Cnidarians exhibit two basic body forms called polyp and medusa. Figure 4.6. The former is a sessile and cylindrical form like Hydra, Adamsia, and so on, whereas the latter is umbrella-shaped and free-swimming like Aurelia or jellyfish. Those cnidarians which exist in both forms exhibit alternation of generations, metagenesis. That is, polyps produce medusae asexually, and medusae form the polyps sexually, for example, Lobelia. Examples: Azalea Portuguese man of war, Dempsia sea anemone, Inatula sea pen, Gorgonia sea fan, and Meandrina brain coral. Figure 4.6. Examples of Coelenterata indicating body forms: Aurelia medusa, E. Adamsia polyp. The figure contrasts the two structural forms found in cnidarians coelenterates. Medusa Aurelia, the jellyfish form, umbrella-shaped, free-swimming, and adapted for locomotion in open water. Tentacles hang downward, equipped with cnidoblasts, specialized stinging cells for prey capture and defense. Polyp Adamsia, a cylindrical sessile form attached to substrates like rocks or shells. Adamsia anemone displays a mutualistic association with hermit crabs, gaining mobility while offering protection to its host. This comparison illustrates the dimorphism of cnidarians, where both forms play distinct ecological roles and can alternate within a single life cycle, a phenomenon called metagenesis. Figure 4.7. Diagrammatic view of cnidoblast. This microscopic diagram depicts a single cnidoblast cell, a defining feature of cnidarians. The cnidoblast contains a capsule called a nematocyst, coiled inside like a harpoon. The trigger cnidocell responds to touch or chemical stimuli, causing the thread to explosively eject, delivering toxins to paralyze prey or deter predators. The cell's nucleus and cytoplasmic components are shown, emphasizing that cnidoblasts are not just defensive structures but also vital for feeding. This figure helps students visualize how such a tiny cellular adaptation has contributed to the ecological success of jellyfish, corals, and sea anemones in marine environments. Phylum: Ctenophora. Ctenophores, commonly known as sea walnuts or cone jellies, are exclusively marine, radially symmetrical, diploblastic organisms with tissue level of organization. The body bears 8 external rows of ciliated cone plates, which help in locomotion, figure 4.8. Digestion is both extracellular and intracellular. Bioluminescence, property of a living organism to emit light, is well marked in ctenophores. Sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external with indirect development. Examples: Pleurobrachia and Ctenoplana. Figure 4.8. Example of Ctenophora pleurobrachia. The figure presents Pleurobrachia, commonly known as the sea walnut, a representative of phylum Ctenophora. The transparent, spherical body is adorned with 8 comb rows, bands of cilia that beat rhythmically to produce a shimmering effect, propelling the animal through water. The diagram shows 2 long retractile tentacles used for capturing planktonic prey. These animals are bioluminescent, producing soft light in the dark ocean through chemical reactions, an adaptation for communication and predator evasion. Ctenophores are important for showing the transition between simple cnidarians and more complex bilaterians, with features like a complete digestive tract and distinct tissues. Phylum: Platyhelminthes. They have dorso-ventrally flattened body, hence are called flatworms, figure 4.9. These are mostly endoparasites found in animals, including human beings. Flatworms are bilaterally symmetrical, triploblastic, and acoelomate animals with organ level of organization. Hooks and suckers are present in the parasitic forms. Some of them absorb nutrients from the host directly through their body surface. Specialized cells called flame cells help in osmoregulation and excretion. Sexes are not separate. Fertilization is internal, and development is through many larval stages. Some members, like Planaria, possess high regeneration capacity. Examples: Tania tapeworm, Fasciola liver fluke. Figure 4.9. Examples of Platyhelminthes are tapeworm bee, liver fluke. This figure illustrates parasitic flatworms, highlighting their dorso-ventrally flattened bodies and adaptations for a parasitic lifestyle. Tapeworm, Tania, a long ribbon-like worm consisting of repeated segments called proglottids, each containing male and female reproductive organs. The scolex head bears suckers and hooks for attachment to the intestinal wall of its host. Liver fluke fasciola, leaf-shaped and hermaphroditic, it lives in the bile ducts of cattle and sheep. It requires 2 intermediate hosts, a snail and an aquatic plant, for completing its life cycle. The figure emphasizes the loss of digestive and sensory systems in favor of reproductive and attachment structures, clear adaptations to endoparasitic existence. Phylum: Aschelminthes. The body of the Aschelminthes is circular in cross-section. Hence, the name roundworms, figure 4.10, they may be free-living, aquatic and terrestrial, or parasitic in plants and animals. Roundworms have organ system level body organization. They are bilaterally symmetrical, triploblastic, and pseudocolumate animals. Alimentary canal is complete with a well-developed muscular pharynx. An excretory tube removes body wastes from the body cavity through the excretory pore. Sexes are separate dioecious. As males and females are distinct. Often females are longer than males. Fertilization is internal, and development may be direct. The young ones resemble the adult or indirect. Examples: Ascaris roundworm, Wuchereria filaria worm, Ancylostoma hookworm. Figure 4.10 dames. Example of Achelminthes, roundworm Ascaris. This figure shows the general structure of Ascaris lumbricoides, a parasitic roundworm belonging to the phylum Achelminthes nemathelminthes. Its body is elongated, cylindrical, and tapered at both ends, covered by a tough cuticle that protects it from digestive enzymes of the host. The sexes are separate dioecious, a key feature distinguishing this phylum. Males are smaller, with a curved posterior end, while females are larger and straight. The figure highlights a complete digestive system with distinct mouth and anus, marking an evolutionary step from incomplete to complete digestion. These worms inhabit the human intestine, causing ascariasis, and represent the advancement toward body organization with a pseudocoelom and distinct organ systems. Phylum, Annelida. They may be aquatic, marine, and freshwater, or terrestrial, free-living, and sometimes parasitic. They exhibit organ system level of body organization and bilateral symmetry. They are triploblastic, metamerically segmented, and chelomate animals. Their body surface is distinctly marked out into segments or metameres and, hence, the phylum name, Annelida Latin. Annulus, little ring, figure 4.11. They possess longitudinal and circular muscles which help in locomotion. Aquatic annelids like Nereis possess lateral appendages, parapodia, which help in swimming. Closed circulatory system is present. Nephridia sing. Nephridium help in osmoregulation and excretion. Neural system consists of paired ganglia sing. Ganglion connected by lateral nerves to a double ventral nerve cord. Nereis, an aquatic form, is dioecious, but earthworms and leeches are monoecious. Reproduction is sexual. Examples: Nereis, Baratima earthworm, and Hirudinaria blood, sucking leech. Figure 4.11. Examples of Annelida. Nereis b. Hirudinaria. This figure contrasts two annelids, marine and parasitic, showing the versatility of segmented worm phylum Annelida. Nereis, free-living marine worm, exhibiting metameric segmentation, the division of the body into ring-like segments both externally and internally. It possesses paired lateral appendages called parapodia for locomotion and respiration. Presence of closed circulatory system and well-developed nervous system indicates a higher level of organization. Hirudinaria leech, an ectoparasite found in fresh water, feeding on the blood of vertebrates. The figure highlights its suckers at both ends and the secretion of hirudin, an anticoagulant preventing blood clotting. Together, they illustrate the adaptive diversity within annelids, from active swimmers to parasitic forms, linked by their shared segmented body plan. Phylum Arthropoda. This is the largest phylum of Animalia, which includes insects. Over 2/3 of all named species on Earth are arthropods. Figure 4.12. They have organ system level of organization. They are bilaterally symmetrical, triploblastic, segmented, and chelomate animals. Body of arthropods is covered by chitinous exoskeleton. The body consists of head, thorax, and abdomen, and have jointed appendages, arthros joint, poda appendages. Respiratory organs are gills, book lungs, or tracheal system. Circulatory system is of open type. Sensory organs like antennae, eyes compound and simple. Statocysts or balancing organs are present. Excretion takes place through Malpighian tubules. They are mostly dioecious. Fertilization is usually internal. They are mostly oviparous. Development may be direct or indirect. Examples: economically important insects: Apis honeybee, bombyx silkworm, Laxifer lac, insect vectors, Anopheles, culex, and Aedes mosquitoes. Gregarious pest: Locusta locust. Living fossil: Limulus king crab. Figure 4.12. Examples of arthropoda: a locust bee, butterfly C, scorpion D, prawn. This figure showcases representatives of the largest animal phylum, Arthropoda, distinguished by jointed appendages and a chitinous exoskeleton. Locust, a terrestrial insect with 3 distinct body regions: head, thorax, abdomen, and wings. Its ability to form swarms demonstrates social and behavioral adaptations. Butterfly displays striking metamorphosis from larva to adult, an adaptation for exploiting different habitats and food sources during life stages. Scorpion, an arachnid with segmented tail ending in a venomous sting, adapted for predation and defense. Prawn, crustacean with gills, for respiration and appendages specialized for swimming and feeding. Collectively, the figure represents the extraordinary adaptive radiation of arthropods in air, land, and water, making them the most successful group in the animal kingdom. Phylum, Mollusca. This is the second largest animal phylum. Figure 4.13. Mollusks are terrestrial or aquatic, marine, or freshwater, having an organ system level of organization. They are bilaterally symmetrical, triploblastic, and coelomate animals. Body is covered by a calcareous shell and is unsegmented with a distinct head, muscular foot, and visceral hump. A soft and spongy layer of skin forms a mantle over the visceral hump. The space between the hump and the mantle is called the mantle cavity, in which feather-like gills are present. They have respiratory and excretory functions. The anterior head region has sensory tentacles. The mouth contains a file-like rasping organ for feeding, called radula. Figure 4.13. Examples of mollusca. Pila B. octopus. This figure highlights the diversity of soft-bodied animals under phylum Mollusca. Pila apple snail is a coiled shell and a soft, unsegmented body divided into head, muscular foot, and visceral hump. The mantle secretes the shell and encloses the mantle cavity, which houses gills syningia for respiration. Octopus, a shellless cephalopod showing extreme adaptation for an active predatory lifestyle. It has 8 muscular arms lined with suckers, a large brain, and well-developed eyes, signs of remarkable cephalization. This figure demonstrates how mollusks evolved from slow-moving shelled forms to intelligent, agile predators, representing a leap in neural and behavioral sophistication among invertebrates. They are usually dioecious and oviparous, with indirect development. Examples: Gila apple snail, Punctata pearl oyster, sepia cuttlefish, loligo squid, octopus devilfish, Plesia sea hare, Dentalium tusk shell, and Chaetoplura chitin. Phylum Echinodermata. These animals have an endoskeleton of calcareous ossicles and, hence, the name Echinodermata spiny-bodied. Figure 4.14. All are marine with organ system level of organization. The adult echinoderms are radially symmetrical, but larvae are bilaterally symmetrical. They are triploblastic and chelomate animals. Digestive system is complete with mouth on the lower ventral side and anus on the upper dorsal side. The most distinctive feature of echinoderm is the presence of water vascular system, which helps in locomotion, capture, and transport of food and respiration. An excretory system is absent. Sexes are separate. Reproduction is sexual. Fertilization is usually external. Development is indirect with free-swimming larvae. Examples: Asterias starfish, Echinus sea urchin, Antedon sea lily, Hookamaria sea cucumber, and Ophiura brittle star. Figure 4.14. Examples of Echinodermata. Asterias B. Ophiura. This figure portrays typical members of phylum Echinodermata, which are exclusively marine and display radial symmetry in adults, bilateral in larvae. Asterias starfish has a 5-armed body radiating from a central disc. The figure emphasizes its water vascular system, used for locomotion, food capture, and respiration. Tiny tube feet protrude through the arms, enabling slow, powerful movement. Ophiura brittle star resembles a starfish but has slender, flexible arms distinctly set off from the central disc, adapted for quick, snake-like movements. The figure underscores echinoderm's unique features: endoskeleton of calcareous ossicles and regenerative ability that distinguish them as the most advanced invertebrates and close evolutionary relatives of chordates. Phylum Hemichordata. Hemichordata was earlier considered as a subphylum under phylum Chordata, but now it is placed as a separate phylum under non-Chordata. Hemichordates have a rudimentary structure in the collar region called stomochord, a structure similar to notochord. This phylum consists of a small group of worm-like marine animals with organ system level of organization. They are bilaterally symmetrical, triploblastic, and collimate animals. The body is cylindrical and is composed of an anterior proboscis, collar, and a long trunk, figure 4.15. Circulatory system is of open type. Respiration takes place through gills. Excretory organ is proboscis gland. Sexes are separate. Fertilization is external. Development is indirect. Examples: balanoglossus and saccoglossus. Figure 4.15. Balanoglossus. This figure depicts balanoglossus, commonly called the acorn worm, which belongs to the phylum Hemichordata, a group that bridges non-chordates and chordates. The body is divided into 3 distinct regions. Proboscis, a muscular, conical structure that helps in burrowing and fruit collection. Collar connects the proboscis and trunk, containing a short portion of the gut and the mouth. Trunk, the elongated posterior part housing the pharynx, gill slits, and reproductive organs. The figure emphasizes its gill slits and notochord-like structure restricted to the proboscis region, highlighting why hemichordates were once considered primitive chordates. It shows the evolutionary significance of balanoglossus as a connecting link between invertebrates and vertebrates, possessing both simple body organization and early chordate-like features. Chordata. Animals belonging to phylum Chordata are fundamentally characterized by the presence of a notochord, a dorsal hollow nerve cord, and paired pharyngeal gill slits, figure 4.16. These are bilaterally symmetrical, triploblastic coelomate with organ system level of organization. They possess a postanal tail and a closed circulatory system. Figure 4.16. Chordata characteristics. This diagram summarizes the 3 diagnostic features that define members of phylum Chordata. Notochord, a flexible, rod-like structure running along the body, providing skeletal support and serving as a primitive backbone in chordates. Dorsal hollow nerve cord, positioned above the notochord. It later develops into the brain and spinal cord in higher vertebrates. Pharyngeal gill slits, openings in the pharynx that function in filter feeding or respiration at some stage of development. The figure also indicates the postanal tail, which aids in movement and balance, and the endostyle or thyroid gland involved in filter feeding and metabolism. This schematic representation unifies all chordates, from simple amphioxus to humans, under a shared structural blueprint that has been modified through evolution for different lifestyles. Table 4.1. Comparison of chordates and non-chordates. This table contrasts the key structural and functional differences between chordates and non-chordates in a point-by-point manner. First, notochord, present in chordates at least in the embryonic stage, but absent in non-chordates. Second, nerve cord, dorsal and hollow in chordates, ventral and solid in non-chordates. Third, circulatory system, closed in chordates, usually open in non-chordates. Fourth, heart position, ventral in chordates, dorsal or absent in non-chordates. And last, gill slits and post-anal tail, present in chordates at some life stage, absent in non-chordates. This comparison table provides a comprehensive evolutionary perspective, showing how the emergence of the notochord and dorsal nerve cord marked the beginning of vertebrate lineage and greater physiological specialization. Phylum Chordata is divided into three subphyla: Urochordata or Tunicata, Cephalochordata, and Vertebrata. Subphyla Urochordata and Cephalochordata are often referred to as protochordates figure 4.17 and are exclusively marine. In Urochordata, notochord is present only in larval tail, while in Cephalochordata it extends from head to tail region and is persistent throughout their life. Examples: Urochordata, Cydia, Salpa, Doliolum, Cephalochordata, Branchiostoma, Amphioxus, or Lancelet. The members of subphylum Vertebrata possess notochord during the embryonic period. The notochord is replaced by a cartilaginous or bony vertebral column in the adult. Thus, all vertebrates are chordates, but all chordates are not vertebrates. Besides the basic chordate characters, vertebrates have a ventral muscular heart with 2, 3, or 4 chambers, kidneys for excretion and osmoregulation, and paired appendages which may be fins or limbs. Figure 4.17. Cydia. The figure shows a Cydia sea squirt, a urochordate belonging to subphylum Tunicata. The adult form is sessile and sac-like, encased in a tunic made of tunicin, a cellulose-like substance. It has 2 siphons, incurrent and excurrent, for water flow through the body. Although the adult lacks a notochord and nerve cord, the larval stage possesses both, along with a post-anal tail. This feature demonstrates the retrogressive metamorphosis, where the larva shows advanced chordate traits that are lost in the sedentary adult. The figure helps students understand the evolutionary reduction and specialization within chordates, emphasizing how developmental stages can reveal ancestral characteristics. Narration for the subphylum Vertebrata classification diagram. This diagram presents a clear overview of the classification of the subphylum Vertebrata, showing the evolutionary division based on the presence or absence of jaws and limbs. At the top, we have Vertebrata, which is divided into 2 major groups, Agnatha and Gnathostomata. 1. Agnatha. These are jawless vertebrates. The term agnatha literally means without jaws. They represent the most primitive vertebrates, lacking a true jaw structure. The diagram shows that agnatha contains only one class, Cyclostomata. Cyclostomes are eel-like, parasitic or free-living fishes such as lampreys and hagfishes. They possess a circular sucking mouth, a cartilaginous skeleton, and gill slits for respiration. 2. Gnathostomata. These vertebrates are characterized by the presence of jaws, an evolutionary advancement allowing more efficient feeding and predation. Gnathostomata is further divided into 2 major groups, Pisces and Tetrapoda, classified here as superclasses. Pisces fishes. These are aquatic vertebrates that bear fins. The diagram indicates 2 classes within Pisces. First, Chondrichthyes, cartilaginous fishes like sharks, rays, and skates, possessing a skeleton made of cartilage instead of bone. Second, Ostectides, bony fishes like Catla, Hippocampus, and others which have a bony endoskeleton, scales, and a swim bladder for buoyancy. Tetrapoda. This group includes vertebrates that bear limbs, enabling life on land or adaptations for terrestrial locomotion. Tetrapods are divided into 4 classes. First, amphibia, vertebrates like frogs and salamanders, which exhibit dual life, beginning in water and maturing on land, possessing moist skin for respiration. Second, reptilia, Reptiles such as snakes, lizards, turtles, and crocodiles, adapted for life on land with scaly skin, internal fertilization, and amniotic eggs. Third, aves, birds characterized by feathers, beaks, hollow bones, and adaptations for flight. Fourth, mammals, warm-blooded vertebrates with hair, mammary glands for feeding young, and highly developed nervous and circulatory systems. This hierarchical diagram efficiently summarizes the evolutionary progression in vertebrates, from primitive jawless forms to advanced jawed, finned, and limbed vertebrates, highlighting how the acquisition of jaws and limbs shaped the diversity of life on Earth. Class Cyclostomata. All living members of the class Cyclostomata are ectoparasites on some fishes. They have an elongated body bearing -6 pairs of gill slits for respiration. Cyclostomes have a sucking and circular mouth without jaws. Figure 4.18. Their body is devoid of scales and paired fins. Cranium and vertebral column are cartilaginous. Circulation is of closed type. Cyclostomes are marine but migrate for spawning to fresh water. After spawning, within a few days, they die. Their larvae, after metamorphosis, return to the ocean. Examples: Petromyzon lamprey and myxine hagfish. Figure 4.18, a jawless vertebrate, Petromyzon. This figure represents Petromyzon lamprey, a jawless fish from the class Cyclostomata, the most primitive group of living vertebrates. The eel-like body is elongated, scaleless, and lacks paired fins. The circular suctorial mouth functions as an attachment organ, helping it cling to other fish and feed on their body fluids. The figure highlights the 7 pairs of gill openings, cartilaginous skeleton, and notochord persisting throughout life, features that signify its primitive vertebrate nature. Petromyzon bridges the evolutionary gap between invertebrates and jawed vertebrates, showcasing how early vertebrates evolved fundamental traits like a cranium and closed circulatory system before the evolution of jaws and paired limbs. Class Chondrichthyes. They are marine animals with streamlined body and have cartilaginous endoskeleton. Figure 4.19. Mouth is located ventrally. Notochord is persistent throughout life. Gill slits are separate and without operculum gill cover. Skin is tough, containing minute placoid scales. Teeth are modified placoid scales which are backwardly directed. Their jaws are very powerful. These animals are predaceous. Due to the absence of air bladder, they have to swim constantly to avoid sinking. Figure 4.19. Examples of cartilaginous fishes. Ascoliodon B. pristis. This figure represents 2 members of the class Chondrichthyes, also known as cartilaginous fishes, characterized by a skeleton made entirely of cartilage. Ascoliodon dogfish exhibits a streamlined, spindle-shaped body adapted for active swimming. The figure highlights paired fins, a heterocercal, unequal-lobed tail for propulsion, and 5 pairs of gill slits without an operculum. Its mouth is ventrally placed and skin is covered with placoid scales which reduce friction and offer protection. B. pristis sawfish. Noted for its elongated snout with tooth-like projections resembling a saw, used for defense and stirring up prey. Both examples demonstrate the adaptive evolution of primitive vertebrates for aquatic predation, possessing cartilaginous endoskeletons, powerful jaws, and well-developed sensory organs, but lacking a swim bladder. Hence, they must swim continuously to avoid sinking. Heart is 2-chambered, 1 auricle, and 1 ventricle. Some of them have electric organs, for example, torpedo, and some possess poison sting, for example, trigon. They are cold-blooded poikilothermous animals. That is, they lack the capacity to regulate their body temperature. Sexes are separate. In males, pelvic fins bear claspers. They have internal fertilization, and many of them are viviparous. Examples: Goliodon dogfish, pristis sawfish, Parcharodon, great white shark, trigon stingray. Class Osteichthyes. It includes both marine and freshwater fishes with bony endoskeleton. Their body is streamlined. Mouth is mostly terminal, figure 4.20. They have 4 pairs of gills, which are covered by an operculum on each side. Skin is covered with cycloid ctenoid scales. Air bladder is present, which regulates buoyancy. Heart is 2-chambered, 1 auricle, and 1 ventricle. They are cold-blooded animals. Sexes are separate. Fertilization is usually external. They are mostly oviparous and development is direct. Examples: marine, Exochetus flying fish, hippocampus, seahorse, freshwater, labeo rohu, catla, Clarius mager, aquarium, betta fighting fish, pterophyllum angelfish. Figure 4.20 examples of bony fishes, a hippocampus catla. This figure showcases members of the class Osteichthyes, or bony fishes, which are characterized by a bony endoskeleton, cycloid or ctenoid scales, and an operculum covering gill slits. The hippocampus seahorse possesses an upright posture and curved tail for clinging to vegetation. Interestingly, in this species, males carry the fertilized eggs in a brood pouch until hatching, a rare reproductive adaptation. B. Catla, freshwater fish commonly found in Indian rivers. Known for its deep body and large head. It has a terminal mouth, operculum-covered gills, and a swim bladder, helping it maintain buoyancy. The figure emphasizes how bony fishes display enormous diversity and advanced features such as air bladder regulation, external fertilization, and streamlined bodies suited for rapid movement and stability in various aquatic environments. Class Amphibia. As the name indicates, gender, amphi, dual, bios, life, amphibians can live in aquatic as well as terrestrial habitats. Figure 4.21. Most of them have 2 pairs of limbs. Body is divisible into head and trunk. Tail may be present in some. The amphibian's skin is moist without scales. The eyes have eyelids. A tympanum represents the ear. Alimentary canal. Urinary and reproductive tracts open into a common chamber called cloaca, which opens to the exterior. Respiration is by gills, lungs, and through skin. The heart is 3-chambered, 2 auricles, and 1 ventricle. These are cold-blooded animals. Sexes are separate. Fertilization is external. They are oviparous and development is indirect. Examples: bufo toad, rana frog, hyla tree frog, salamandra salamander, ichthyophis limbless amphibia. Figure 4.21. Examples of amphibia. A salamandra v. rana. This figure depicts members of class Amphibia, representing the first vertebrates to live both in water and on land. A salamandra, salamander, a lizard-like amphibian showing tail retention in adults and smooth, moist skin essential for cutaneous respiration. B. Rana frog, common amphibian adapted for dual life. Its webbed feet aid in swimming, while long hind limbs support leaping on land. The figure demonstrates how amphibians bridge aquatic and terrestrial habitats, with features such as 3-chambered hearts, glandular skin for respiration, and external fertilization. Their life cycle includes metamorphosis, transitioning from gill-breathing larvae to air-breathing adults, marking a major evolutionary leap toward terrestrial life. Class Reptilia. The class name refers to their creeping or crawling mode of locomotion. Latin repere or reptum, creep or crawl. They are mostly terrestrial animals and their body is covered by dry and cornified skin, epidermal scales or scutes, figure 4.22. They do not have external ear openings. Tympanum represents ear. Limbs, when present, are 2 pairs. Heart is usually 3-chambered, but 4-chambered in crocodiles. Reptiles are poikilotherms. Snakes and lizards shed their scales as skin cast. Sexes are separate. Fertilization is internal. They are oviparous and development is direct. Examples: Cologne turtle, testudo tortoise, chameleon tree lizard, Pilotes garden lizard, Crocodilus crocodile, alligator alligator, Hemidactylus wall lizard, poisonous snakes, nausea cobra, Bangurus crite, vipera viper. Figure 4.22. Reptiles, chameleon bee, Crocodilus C, Chelone D, nausea. This figure exhibits diverse members of class Reptilia, which evolved complete adaptation to land through scaly, water-resistant skin and internal fertilization. A chameleon, arboreal reptile with a prehensile tail, independently movable eyes, and color-changing skin, adaptations for camouflage and hunting. Crocodilus, a semi-aquatic reptile with a strong tail and bony scutes. It exhibits four-chambered heart and parental care, both advanced reptilian traits. C. Cologne turtle. Possesses a bony shell carapace and plastron enclosing the body, offering protection. D. Naja cobra. A venomous snake capable of spreading its hood when threatened, used for both defense and intimidation. Together, the figure shows the evolutionary diversification of reptiles, with physiological adaptations like lungs for respiration, amniotic eggs, and ectothermic regulation, allowing them to thrive in terrestrial habitats. Class Aves. The characteristic features of Aves birds are the presence of feathers, and most of them can fly except flightless birds, for example, ostrich. They possess beak, figure 4.23. The forelimbs are modified into wings. The hind limbs generally have scales and are modified for walking, swimming, or clasping the tree branches. Skin is dry, without glands except the oil gland at the base of the tail. Endoskeleton is fully ossified bony, and the long bones are hollow with air cavities pneumatic. The digestive tract of birds has additional chambers, trap and gizzard. Heart is completely four-chambered. They are warm-blooded homoeothermous animals. That is, they are able to maintain a constant body temperature. Respiration is by lungs. Air sacs connected to lungs supplement respiration. Sexes are separate. Fertilization is internal. They are oviparous, and development is direct. Examples: Orvis crow, Palumba pigeon, Psittacula parrot, Druthio ostrich, Avo peacock, Captain Heights penguin, Neophron vulture. Figure 4.23. Some birds, a Neophron bee, Druthio C. Psittacula D. pavo. This figure presents representatives of class Aves, highlighting their structural adaptations for aerial life. A Neophron vulture, a scavenger bird with broad wings for soaring flight and a strong beak for tearing flesh. B. Struthio ostrich, the largest living bird, flightless but specialized for running, with strong legs and reduced wings. Psittacula parrot possesses a curved beak and zygodactyl feet for climbing and grasping, known for intelligence and vocal mimicry. D. Pavo peacock, national bird of India, noted for its brilliant plumage and ornamental tail feathers used in courtship displays. The figure collectively illustrates avian features: feathers, light bones, wings, and warm-blooded metabolism, which evolved for efficient flight and thermoregulation, marking birds as highly specialized vertebrates. Class Animalia. They are found in a variety of habitats: polar ice caps, deserts, mountains, forests, grasslands, and dark caves. Some of them have adapted to fly or live in water. The most unique mammalian characteristic is the presence of milk-producing glands, mammary glands, by which the young ones are nourished. They have two pairs of limbs, adapted for walking, running, climbing, burrowing, swimming, or flying. The skin of mammals is unique in possessing hair. External ears or pinnae are present. Different types of teeth are present in the jaw. Heart is four-chambered. They are homoeothermous. Respiration is by lungs. Sexes are separate, and fertilization is internal. They are viviparous with few exceptions, and development is direct. Examples: Oviparous Ornithorhynchus platypus, Viviparous Macropus kangaroo, Pteropus, flying fox, camelus camel, macaca monkey, ratus rat, canis dog, phallus cat, elephas elephant, equus horse, delphinus common dolphin, Balanoptera blue whale, Panthera tigris tiger, Panthera Leo lion. Figure 4.24. Some mammals: A. Ornithorhynchus, B. Macropus, C. Pteropus, D. Balanoptera. This figure illustrates the diversity and adaptive specialization within class Mammalia, the most advanced class of vertebrates. The Ornithorhynchus platypus, monotreme or egg-laying mammal. It exhibits primitive mammalian features like egg-laying and cloaca, alongside advanced traits like mammary glands for lactation. The duck-like bill is electroreceptive, helping it locate prey underwater. B. Macropus kangaroo, a marsupial that carries young in a ventral pouch. Adapted for terrestrial life, it has powerful hind limbs for hopping and long tail for balance. This figure demonstrates reproductive specialization and locomotor adaptation. C. Pteropus, flying foxbat, placental mammal capable of powered flight. The wings are modified forelimbs, while the body is covered with fur for insulation. The figure highlights the ecological role of bats in pollination and seed dispersal, showing behavioral and anatomical adaptations. D. Balanoptera blue whale represents aquatic mammals with adaptations for marine life, including streamlined body, blubber for insulation, and modified forelimbs as flippers. The whale shows evolutionarily advanced placental reproduction and complex social behavior, bridging terrestrial and aquatic adaptations in mammals. This figure collectively emphasizes mammalian diversity, highlighting reproductive strategies, locomotion, feeding adaptations, and ecological niches, marking mammals as highly evolved vertebrates with complex physiology and behavior. The salient distinguishing features of all phyla under Animal Kingdom is comprehensively given in the Table 4.2. Table 4.2. Salient features of different phyla in the Animal Kingdom. Let's explore the major animal phyla and their distinguishing characteristics, as summarized in this table. We start with the simplest animals and move toward the more complex forms, highlighting their organization, symmetry, body cavity, and unique adaptations. Porifera fungus. These are the most primitive multicellular animals. They are asymmetrical or sometimes show radial symmetry. Their bodies are cellular level of organization without true tissues or organs. Porifera are acoelomate, meaning they have no body cavity. They live mostly in marine environments and are sessile filter feeders, drawing water through pores to obtain food. Palanturata cnidarians. These animals exhibit radial symmetry and have a tissue-level organization with two germ layers, ectoderm and endoderm. They are diploblastic and acoelomate, possessing a gastrovascular cavity for digestion. Coelenterates include hydra, jellyfish, and corals, and they have cnidoblasts, specialized stinging cells used for defense and prey capture. Platyhelminthes, flatworms. Platyhelminths are bilaterally symmetrical and triploblastic, showing organ-level organization. They are acoelomate, lacking a body cavity between the gut and body wall. Examples include planaria, tapeworms, and liver flukes. They exhibit cephalization, primitive digestive system, and adaptations for both free-living and parasitic lifestyles. Aschelminthes, or nematode roundworms. Roundworms are bilaterally symmetrical and triploblastic, with pseudocoelom, body cavity not fully lined with mesoderm. Their bodies are elongated and cylindrical with a complete digestive system. Many, like askers, are parasitic, while others are free-living. They have separate sexes and exhibit simple excretory and nervous systems. Annelida segmented worms. Annelids show bilateral symmetry, triploblastic organization, and are chelomates, meaning they possess a true body cavity lined with mesoderm. Their bodies are metamerically segmented, each segment containing repeated organs and structures. Examples include earthworms, leeches, and nereis. Annelids have a closed circulatory system, a well-developed nervous system, and excretory structures called nephridia. Arthropoda insects, crustaceans, and arachnids. This largest animal phylum exhibits bilateral symmetry, triploblastic organization, and chylomate body cavity. Arthropods have a chitinous exoskeleton, jointed appendages, and segmented bodies. Examples include insects, scorpions, crabs, and prawns. They have a well-developed nervous system, compound eyes, and exhibit remarkable adaptations for terrestrial, aerial, and aquatic habitats. Mollusca snails, octopus, clams. Mollusks are bilaterally symmetrical, triploblastic, and chylomate. They have a soft, unsegmented body divided into head, foot, and visceral mass, often protected by a calcareous shell. Examples include Gila, octopus, and clams. They possess a mantle, complete digestive system, open or closed circulatory system, and well-developed sensory organs in some species. Echinodermata, starfish. Sea urchin. Echinoderms are radially symmetrical as adults, but their larvae are bilaterally symmetrical, reflecting evolutionary transition. They are triploblastic and coelomate, with a calcareous endoskeleton and a unique water vascular system used for locomotion and feeding. Examples include Asterias and Ophiura. They have a complete digestive system, lack excretory organs, and show remarkable regeneration ability. Hemichordata balanoglossus. Hemichordates are bilaterally symmetrical, triploblastic, and chylomate. They show a dorsal nerve cord and gill slits. The notochord is limited to the proboscis region. Balanoglossus is a burrowing marine worm demonstrating a combination of invertebrate and chordate features, representing an evolutionary link. Chordata fishes, amphibia, reptiles, birds, mammals. Chordates are bilaterally symmetrical, triploblastic, and chylomate, characterized by a notochord, dorsal hollow nerve cord, pharyngeal gill slits, postanal tail, and endostyle or thyroid gland. Subphyla include Urochordata, Cephalochordata, and Vertebrata. Vertebrates exhibit complex organ systems, advanced nervous and circulatory systems, and adaptations to diverse habitats, ranging from aquatic to terrestrial and aerial environments. This table gives us a clear overview of the evolutionary progression of animals, showing how structural complexity, body cavity development, symmetry, and organ systems evolved from simple sponges to highly specialized vertebrates. Summary. The basic fundamental features such as level of organization, symmetry, cell organization, coelom, segmentation, notochord, and so on, have enabled us to broadly classify the animal kingdom. Besides the fundamental features, there are many other distinctive characters which are specific for each phyla or class. Porifera includes multicellular animals which exhibit cellular level of organization and have characteristic flagellated choanocytes. The cheilenterates have tentacles and bear cnidoblasts. They are mostly aquatic, sessile, or free-floating. The ctenophores are marine animals with comb plates. The platyhelminths have flat body and exhibit bilateral symmetry. Parasitic forms show distinct suckers and hooks. Aschelminthes are pseudocoelomates and include parasitic as well as non-parasitic roundworms. Annelids are metamerically segmented animals with a true coelom. The arthropods are the most abundant group of animals characterized by the presence of jointed appendages. The mollusks have a soft body surrounded by an external calcareous shell. The body is covered with external skeleton made chitin. The echinoderms possess a spiny skin. Their most distinctive feature is the presence of water vascular system. The hemichordates are a small group of worm-like marine animals. They have a cylindrical body with proboscis, collar, and trunk. Phylum Chordata includes animals which possess a notochord either throughout or during early embryonic life. Other common features observed in the chordates are the dorsal, hollow nerve cord, and paired pharyngeal gill slits. Some of the vertebrates do not possess jaws, agnatha, whereas most of them possess jaws, gnatha, stomata. Agnatha is represented by the class Cyclostomata. They are the most primitive chordates and are ectoparasites on fishes. Gnathostomata has two superclasses, Pisces and Tetrapoda. Classes Chondrichthyes and Osteichthyes bear fins for locomotion and are grouped under Pisces. The Chondrichthyes are fishes with cartilaginous endoskeleton and are marine. Classes Amphibia, Reptilia, Aves, and Mammalia have two pairs of limbs and are thus grouped under Tetrapoda. The amphibians have adapted to live both on land and water. Reptiles are characterized by the presence of dry and cornified skin. Limbs are absent in snakes. Fishes, amphibians, and reptiles are poikilothermous, cold-blooded. Aves are warm-blooded animals with feathers on their bodies and forelimbs modified into wings for flying. Hind limbs are adapted for walking, swimming, perching, or clasping. The unique features of mammals are the presence of mammary glands and hairs on the skin. They commonly exhibit viviparity. You've just finished listening to Chapter 4, Animal Kingdom from NCERT, Class 11 Biology, here on Biodecode. This is Biodecode, and you're listening to Chapter 4, Animal Kingdom, Exercise Solutions and Key Questions. Question 1. What are the difficulties that you would face in classification of animals if common fundamental features are not taken into account? Answer. Firstly, without considering the common fundamental features, it would become extremely difficult to categorize animals into coherent groups. Classification relies on identifying shared characteristics such as symmetry, body plan, and embryological development. If these are ignored, unrelated animals might be grouped together, while closely related ones could be separated. Second, it would hinder the understanding of evolutionary relationships and make comparative studies inefficient. Third, scientific communication and documentation would become confusing, as researchers would lack a consistent framework to refer to different groups of animals. Thus, considering fundamental features is essential for a logical, systematic, and meaningful classification of the animal kingdom. Question 2. If you are given a specimen, what are the steps that you would follow to classify it? Answer. Observation begins with examining the overall body structure, symmetry, and segmentation, noting whether the organism has a coelom and what type of body cavity it possesses. Second, one would investigate the organization of tissues, type of digestive system, and the presence or absence of specialized organs, such as respiratory or excretory structures. Third, embryological development patterns, such as direct or indirect development, can provide clues to its taxonomic group. Fourth, one examines skeletal structures, appendages, or external features to determine higher-level classifications. Finally, molecular and genetic studies, when available, confirm the classification, providing a complete and systematic placement of the specimen within the animal kingdom. Question 3. How useful is the study of the nature of body cavity and coelom in the classification of animals? Answer. The study of body cavities is crucial because the presence or absence of a coelom distinguishes major animal groups. For example, acoelomates like flatworms lack a body cavity, while pseudocoelomates such as roundworms have a partially lined cavity, and coelomates like annelids and chordates have a true coelom lined with mesoderm. Second, the type of body cavity influences organ development, body flexibility, and complexity of systems, which are key criteria in classification. Third, coelom structure often reflects evolutionary advancement, allowing taxonomists to infer relationships and lineage among phyla. Overall, understanding the body cavity provides both functional and evolutionary insights, making it a cornerstone in animal taxonomy. Question 4. Distinguish between intracellular and extracellular digestion. Answer. Intracellular digestion occurs inside specialized cells, where food is engulfed by phagocytosis and broken down within vacuoles using enzymes. This type of digestion is typical of simpler animals such as sponges, where a true digestive cavity is absent. Second, extracellular digestion takes place in a digestive cavity or lumen outside the cells, allowing the breakdown of larger food particles. Enzymes secreted into the cavity digest the food externally before absorption. This mechanism is seen in more complex animals like annelids, mollusks, and vertebrates. Third, the distinction lies in the location of digestion and complexity. Intracellular digestion is simpler and cell-based, whereas extracellular digestion supports more complex and larger body plans. Question 5. What is the difference between direct and indirect development? Answer. Direct development refers to a mode where the organism hatches or is born as a miniature version of the adult, without any distinct larval stage. Examples include most reptiles and birds. Second, indirect development involves a larval stage that is morphologically different from the adult and undergoes metamorphosis to reach maturity, as seen in amphibians and many insects. Third, this distinction is crucial in classification and understanding life cycles, ecological adaptations, and evolutionary strategies of different animal groups. Question 6. What are the peculiar features that you find in parasitic platyhelminthes? Answer. Parasitic platyhelminthes exhibit adaptations for a life within the host. Their body is dorsoventrally flattened, allowing them to reside in narrow spaces. Second, they possess a tough outer covering called a tegument, which protects against the host's digestive enzymes and immune responses. Third, they often lack a digestive system entirely, or have a highly reduced one, absorbing nutrients directly from the host's body. Fourth, reproductive capacity is greatly enhanced, with hermaphroditism and prolific egg production ensuring survival and spread. These features collectively represent specialized adaptations for parasitism. Question 7. What are the reasons that you can think of for the arthropods to constitute the largest group of the animal kingdom? Answer. Arthropods have an exoskeleton made of chitin that provides protection, support, and reduces water loss. Second, their segmented body plan with jointed appendages allows great mobility, specialization, and adaptability to diverse habitats. Third, they have a highly developed sensory system, including compound eyes and antennae, which aids in navigation, finding food, and avoiding predators. Fourth, their reproductive strategies, short life cycles, and ability to adapt to environmental changes contribute to their remarkable diversity. Collectively, these factors have enabled arthropods to become the largest and most successful group in the animal kingdom. Question 8. Water vascular system is the characteristic of which group of the following? Porifera, Ctenophora, Echinodermata, Chordata? Answer. The correct answer is Echinodermata. First, this group possesses a unique water vascular system, which consists of canals filled with seawater that help in locomotion, feeding, and gas exchange. Second, tube feet arising from these canals allow the echinoderm to move, capture prey, and adhere to surfaces. Third, this system is absent in Porifera, Ctenophora, and Chordata, making it a distinct feature of echinoderms. Question 9. All vertebrates are chordates, but all chordates are not vertebrates. Justify the statement. Answer. All vertebrates belong to the phylum Chordata because they possess fundamental chordate features such as a notochord, dorsal hollow nerve cord, pharyngeal slits, and post-anal tail at some stage of development. Second, not all chordates are vertebrates because some subphyla such as Urochordata and Cephalochordata, lack a well-developed vertebral column. Third, the vertebral column is the defining characteristic of vertebrates. The basic chordate plan is broader and includes both vertebrates and invertebrate chordates. Question 10. How important is the presence of air bladder in Pisces? Answer. The air bladder, or swim bladder, is essential for maintaining buoyancy in water, allowing fishes to float at different depths without constant swimming. Second, it aids in respiration in some species, functioning as an accessory respiratory organ. Third, in some fishes, It also serves as a resonating chamber for sound production or communication. Overall, the air bladder contributes to survival, energy efficiency, and ecological adaptability in aquatic environments. Question 11. What are the modifications that are observed in birds that help them fly? Answer. Birds have lightweight, hollow bones that reduce body weight without compromising strength. Second, their forelimbs are modified into wings, providing lift and thrust. Third, feathers, especially flight feathers, aid in aerodynamics and temperature regulation. Fourth, birds have a high metabolic rate, powerful muscles, and a rigid skeletal structure that supports sustained flight. Fifth, respiratory adaptations, such as air sacs, ensure a continuous supply of oxygen during flight. Collectively, these modifications enable birds to soar, glide, and maneuver efficiently in the air. Question 12. Could the number of eggs or young ones produced by an oviparous and viviparous mother be equal? Why? Answer. The number of eggs or young produced is usually not equal because of differences in reproductive strategy. Oviparous animals lay eggs outside the body and often produce a large number to increase chances of survival, as many eggs may be lost to predation or environmental hazards. Second, viviparous animals give birth to live young, which develop internally and receive nourishment from the mother, resulting in fewer offspring but higher survival rate. Third, this difference reflects an evolutionary trade-off between quantity and parental investment. Question 13. Segmentation in the body is first observed in which of the following? Platyhelminthes, Eschelminthes, Annelida, Arthropoda? Answer. The correct answer is Annelida. First, segmentation or metamerism refers to the division of the body into repeated units called metameres. Second, each segment in annelids contains components of circulatory, excretory, and nervous systems, allowing greater specialization and flexibility. Third, while arthropods also exhibit segmentation, it is more apparent and functional first in annelids, marking an important evolutionary development. Question 14. Match the following: perculum, parapodia, scales, palm plates, radula, hairs, choanocytes, gill slits with respective animal groups. Answer. Operculum is found in osteichthyes, protecting the gills. Second, parapodia are lateral appendages in Annelida, aiding locomotion and respiration. Third, scales are characteristic of reptilia, providing protection and reducing water loss. Fourth, palm plates are ciliary structures in Stenophora, helping in locomotion. Fifth, radula is a feeding organ in mollusca, scraping or cutting food. Sixth, hairs are found in Mammalia, serving sensory, thermoregulatory, and protective functions. Seventh, choanocytes are specialized collar cells in porifera, aiding in water flow and feeding. Finally, gill slits occur in cyclostomata and chondrichthyes, allowing respiration in aquatic chordates. Question 15. Prepare a list of some animals that are found parasitic on human beings. Answer. Examples include Plasmodium, causative agent of malaria transmitted by mosquitoes. Second, Entamoeba histolytica, responsible for amoebic dysentery inhabiting the human intestine. Third, Ascaris lumbricoides, a roundworm found in the human digestive tract. Fourth, Taenia solium and Taenia saginata, tapeworms inhabiting the intestines. Fifth, Pediculus humanus, or the human louse, which lives on hair and skin. 6. Sarcoptes scabii, mite-causing scabies. These parasitic animals exhibit diverse adaptations for survival within or on the human body. Congratulations! You've just completed listening to Unit 1, Diversity of the Living World, covering Chapters 1 to Chapter 4, along with all NCERT exercise solutions and important questions, here on BioDecode. I hope this audio helped you understand the fundamental principles of biological classification, from the definition of life and taxonomy to the intricate organization of plants and animals. We've explored how living organisms are grouped, how they evolved, and how structure and function connect across different kingdoms of life. These ideas form the foundation for everything that follows in biology, especially Unit 2, where we'll move from classification to structure, exploring the anatomy and morphology of plants and animals. So make sure to follow BioDecode and stay tuned for the next part of this NCERT biology audio series. Keep learning, keep exploring, and I'll meet you in the next audio lecture, Unit 2, Structural Organization in Plants and Animals. And yes, students, don't forget to like, share, and subscribe to BioDecode for more NCERT audiobooks, quick revisions, and neat preparation guides. See you in the next audio.